Good morning, everybody. Today is Tuesday, April 28th. We are going to continue our discussion of the nervous system. We have a little bit more brain physiology to talk about, neurophysiology to talk about. And then we're finally going to get a chance to start talking some anatomy. We will talk about the protections of the brain, the anatomy of the brain, and the anatomy of the spinal cord. So those are all things that we are going to be focusing on and talking about starting today. You still have a number of assignments coming up that are going to be due. One of the big ones is your Physio Exercise 3. There are 9 activities for that. So again, as I've talked about in the past, it isn't hard. It just takes a bit of time. So make sure you're working on that because that is due this Thursday. Also this Thursday, as you can see here, it's going to be a big day because we are going to finally get a chance to do that cranial nerve handout. Again, we will be working on it together. Your textbook does a nice job about talking about these things. So I would strongly encourage you to, even if you don't fill out the handout first, read the part where it talks about the cranial nerve so it'll make some sense and it'll be useful and helpful as we go through that together on Thursday. Next week, 2 more assignments due. Your 15-point nervous system review is going to be due, and today's material will really be the last little bit of information you have to be able to correctly answer those things as we talk about some of the more the anatomy. Because again, remember the point of that is to talk about putting structure, function, and location. We really haven't had a chance to talk too much about locations yet, but we will finally get a chance to talk about that today, and that'll help you complete that. And remember, that review is graded for correctness. Again, for many of the answers, it's 1 word or 2 words or 3 words. There's only a couple questions on there that require a sentence or 2 to be able to correctly answer. So if you want to fill it out on the same sheet, that is fine. But also if you want to just type it out in a Word document as a separate piece of question, you know, question 1, this is the answer, that's fine. However you want to do that is fine. I don't require a second piece of paper for this one because there's so little writing is necessary for this one. And then one last unit review, and that's unit 14 review, and that's going to be due Thursday next week. And then 2 weeks from today, we have our lab and lecture exam. Again, the format for the lecture exam is exactly the same as it was for the last one. The format of the lab exam is to be determined. Like I said, my 431 class has their exam tomorrow, and they're going to do a lab exam where they get all the questions at once. So we'll see if that causes more problems or less problems, and then decide how we handle the rest of the lab exams from there. But either way, it's a format you've already seen. It's either going to be the same as the lecture or the same as the last lab. So that's going to be easy. And then the following week, 3 weeks from today, you have your final exam. It is cumulative, and it is on May 19th. Other than that, I don't know anything about it. So it may be multiple choice. It may be 2 hours like the normal one is, or there may be some adjustment to that. So I'm not sure what that will be, but I'll have a better idea after this weekend probably. And like I said, I do have a review to remind you of the concepts. So maybe we'll post that this weekend as well. But notice you have a whole week between the last lab and lecture exam and the final. And there is a lot of material on the nervous system. It's one of our bigger sections. So I would strongly encourage you to not spend too much time focused on the final yet. Focus instead on the lab and lecture exam. Think of it this way. Your final exam is only 100 points. Your lecture exam is 100 points. Your lab exam is worth, this exam probably has somewhere between, I don't know, 60 and 70 questions on it. So obviously this lab and lecture exam count more than your final exam does. Not that your final exam isn't important, because remember, it can replace your lowest lecture score. But one thing at a time. You've got a whole week to study for that final exam. So I would encourage you strongly to not spend too much time on it yet. All righty. I realize I don't have my chat window up. Let's get that up. Any questions before we get started then? All right, then let's go ahead and get started. I'm going to go ahead and turn off the video. And lowest lecture score, no lab. Oh, yes, that is correct. All the lab exams count. So all the lab exams are all worth different point values, so it isn't practical to be able to replace them. The final, and again, as I said, because of this change in format, I do not know if the final will be a straight replacement of your of a lecture score. We have a lecture exam that is worth less than the other lecture exams. Normally, they're worth all the same amount of points. Normally, I have more control over the final exam and what it actually looks like. So I'm not sure the format. There will be a benefit to being successful on your final exam, but I don't know if it'll be a straight replacement or percentage points or how exactly that will work. But again, I will have a better idea this weekend when I get a chance to spend more time focusing on that. All right, any other questions? All right, let's dive into lecture then. Bring back my annotation tools, put that there. And let's talk about signal transmission. Now, if you think about it, we have, and we can use black, talked a lot about the neuron. We know that out here in the cell body and in the dendrites, we have talked about how it has a normal plasma membrane. It has voltage gated and, pardon me, not voltage gated, it has chemically gated and mechanically gated channels. And we talked about out here on the dendrites and cell bodies, we can produce our graded potentials. We then talked about how those graded potentials are going to spread to our axon hillock. And here in our axon hillock, we talked about how we can produce an action potential. Right. Again, we talked about how here the axon's plasma membrane is specialized, has those voltage-gated sodium and voltage-gated voltage-gated sodium and voltage-gated potassium channels. We talked about the characteristics of those. The sodium open quickly, the potassium open slowly and close as well. The sodium has that second gate, then an activation gate that allows it to have 3 states, open, close, and inactive, and all the things that went along with that, producing it at the axon hillock. And then we also talked about how we can then propagate the action potential. And we even talked about doing it with and without myelin. And that's kind of what we spent the last week talking about. The last little bit that we have to deal with is then our goal is for our neuron to communicate with some target cell, whatever that target cell may happen to be. And luckily, this is the part of the neuron that we're most familiar with because we talked about it in the last section. We know that axon branches to form the axon terminals. And at the axon terminals, there is that specialization, that synaptic end bulb. And so we talked about how that cell communicates with the target cell. We talked about in the case here where the target cell was a muscle cell, more specifically skeletal muscle cell. But the target cell could be anything. As we know, neurons communicate with skeletal muscle, smooth muscle, cardiac muscle, glands, and they also communicate to other neurons. So let's write that down. I see there's a question. Let me finish this thought and then I will answer it. So possible target cells include smooth muscle. Let's sneak this back on here a little bit and then make sure we don't go off the edge. Smooth muscle, cardiac muscle. We already said skeletal muscle, but we'll put it again just so it's all in the right place. Glands, and the most common target cell, which is another neuron. Most neurons communicate to other neurons. All right, now let's take a look at that question. Question from the last lecture. For the second action potential, do we need a stronger stimulus and a higher threshold, or just a stronger stimulus, but the threshold is still the same? Threshold is still the same. How you turn on, so again, if you think about it, and we'll just do it in simple terms. Do a graph here, right? And then this is our threshold, right? So if you think about the neuron, when it is at its resting membrane potential, right, it only has this far it needs to go to be able to reach threshold. So we only have to go from, for instance, negative 70 millivolts to negative 60 millivolts. We only need a 10 millivolt change to reach threshold. However, with our action potential, and let's go ahead and change the color to emphasize this, we have, right, the absolute refractory period, where no matter what we do to the cell, we cannot produce a second action potential. And then what we have is that relative refractory period. Notice during this relative refractory period, and actually I drew that too far. So let's cheat, put orange over the top of it so it goes away. There you go. Here in yellow, this period of time, if we're trying to stimulate the cell and get it to threshold, the problem is the cell is trying to hyperpolarize. So if you pick some random spot on here, like right here, right, at this point, the cell might be at negative 85 millivolts. So if you were to give it that same original 10 millivolt stimulus, a 10 millivolt stimulus, wow, that's huge. Let's erase that. A 10 millivolt change from here isn't going to reach threshold, right? So the fact that the cell is getting more negative means we need a stronger stimulus to reach threshold. Remember, with the relative refractory period, it is possible to get another action potential, but it takes a bigger, stronger stimulus to do it. It's harder to do. The same thing you did the first time won't work the second time. You need a bigger stimulus, a stronger signal to get it there. All right, does that make sense? No, it's not the threshold. For our purposes, again, and this is one of those things where you have to remember, the sky is blue. We are dramatically oversimplifying all of this. Because here's the secret. Not all voltage-gated channels threshold is negative 60. They vary dramatically, and in fact, they're dynamic. They're constantly changing based on environment and other things. So we are doing a simple sky is blue version of this. And the simple sky is blue version of this, threshold is negative 60, period. So your goal is to get to negative 60. Is it possible for the threshold to change? Sure. Is that something we have to worry about? No. The point of the relative refractory period is it is harder to make an action potential. Why it's harder doesn't matter, but it takes a bigger stimulus to reach threshold and to get those cells to fire again. But it isn't because the threshold gets higher. That actually doesn't happen, so that's not correct. It's just harder to reach the threshold. Okay, that makes sense? Cool, awesome, excellent. All right, so back to where we were. Get rid of all that. Any other questions on the last stuff? I love getting questions. I don't mind answering those. But before we get back into the flow, I just want to make sure there isn't anything else that we need to clarify. All right, then, like I said, neurons, we've produced gradient potentials, we've produced action potentials, we've propagated action potentials. So the last thing we have to do to understand the physiology of a neuron is to transmit the signal from that neuron to whatever its target cell is. And the good news is we have some familiarity with this. All right, what happens here between the synapse, synaptic end bulb, and the target cell is something we have already discussed and already learned. So this is not really new material. None of the things we're talking about is new material. This may be a new way of thinking about it, but really there's no new information. You know all of this. And in fact, I'll tell you, there's actually 2 different types of synapses, and you already know them both. So let's clear the pictures, and let's go ahead and talk about that. So again, a synapse is that functional junction between a presynaptic neuron and its target cell. So we can do a more simple drawing of this. I'll just do the circle for the cell body. I will just do a line for the axon and a line for the synaptic end bulb, and it is going to talk to a target cell. As it turns out, there are actually 2 types of synapses. We didn't use the term synapse when talking about these things originally, but it turns out there is indeed 2 different types of synapses, and we have talked about both of them. The first type of synapse is what we call an electrical synapse. If you think way back to the beginning of this class, when we talked about cells and we talked about cell junctions, when we talked about putting cells together, right? One of the things we talked about is there's 3 different types of cell junctions. There are the tight occluding junctions, there are the desmosomes, and the other type of channel, the other type of cell junction we talked about was a gap junction. And in that gap junction, we have, let's emphasize that way, make it solid, a protein that is embedded within the walls of the 2 cells, and it produces a fluid-filled pore between the cells. Oops. Did I spell it correctly? With this electrical synapse, there are some huge advantages to this. One of them is that it's fast. Okay. If something changes inside of this cell, like for instance, there is a huge influx of that positive ion because we've got an action potential going on, well, that sodium ion can flow right through that gap junction from one cell to the next. And so that action potential can spread from one cell to the next. Another huge advantage of it is it doesn't have to just go from left to right. If instead there is a huge influx of sodium in this cell, that sodium can just as easily flow through that fluid-filled pole to the other side. So it's fast. It also allows 2-way communication. Right? So that's something that is a huge advantage to this type of junction. However, as fast and as useful as this is, it is not common in the nervous system. Right? Fast 2-way communication, those definitely sound like things that would be beneficial. But here's the catch. Basically, what happens with these cells are the cells are essentially hardwired together. It allows the cells to work as one, which again sounds like a really useful thing. And some places, like in the smooth muscle of our digestive system, like in the cardiac muscle of our heart, getting all those cells to work together as one, it sounds like a really positive thing. But here's the downside. When things are hardwired, there's no decision making. Right? It's like the light switch on your wall. You flip it up, the light goes on. You flip it down, light goes off. And you could sit here all day, every day, flipping that switch on and on and on and on. And at no point when you flip that switch is the window going to open, or the garbage disposal downstairs is going to go on, right? Or the light in another bedroom goes on or anything like that. There's no decision making. It's hardwired together. And while it is important for the cells in our nervous system to communicate rapidly, to work together as one and things along those lines, we also want them to be modifiable. We want to be able to make decisions. We want to be able to change our behavior. I already used the example, me sitting here with my microphone talking to you across the computer, and I say to you in my biggest, scariest voice, I'm going to kill you. When I say I'm going to kill you, you're not going to respond to that. But like I said, a bear with an axe busts in your door, stares you in the eye, and says, I'm going to kill you. And instead, you are going to get a much more different response as a result of that. All right? And so we want to be able to make decisions. We want to be able to modify our behavior. And an electrical synapse will not allow us to do that. So there's some huge advantages to this. All right, let me go ahead and clear this. Like I said, it's fast. It allows for that two-way transmission of information, and it allows the cells to work together as one. But it's not common in the nervous system. It's found more in smooth muscle. It's found more in cardiac muscle and locations like that, where we want these big, large, smooth muscle walls or the heart of the cardiac muscle of the heart to work together as one functional unit. Instead, we want to be able to make decisions. And the way you make decisions is by using a chemical synapse. And a chemical synapse is something that we have already seen and we have already talked about. We know that the chemical synapse has a specialization to the, and again now, no new information, but we are going to add a little vocabulary. There is a specialization to the neuron that is communicating with the target cell, and we call that the presynaptic cell. Again, that's just a fancy way of saying the neuron. There's a specialization to the presynaptic cell. That specialization to the presynaptic cell, to the neuron, as we know, is the synaptic end bulb. And thankfully, we already know what's special about it. It basically has two big specializations. One of those are vesicles, and those vesicles contain a chemical that is going to be used for communication. And of course, what is the generic term for the chemical that is used for communication? Well, neurotransmitter is the general, absolutely. Acetylcholine is a specific type of neurotransmitter. But the generic term, the general term is a neurotransmitter. Acetylcholine is the most common neurotransmitter in the peripheral nervous system, but it is a specific example. And of course, we want to control the release of that. And we do that by having those specialized channels on our synaptic end bulb. And of course, those specialized channels are the voltage gated calcium channels. All right, because if we want a cell to do a wonky thing, and releasing acetylcholine or neurotransmitter is a wonky thing, we need calcium to be able to do it. Sorry, I left my door of my office open and my daughters were giving me sour looks for making too much noise. All right. We know that this needs to communicate with our target cell, and usually some specialization like for instance, our motor end plate that we talked about, we're gonna need some specialization to that target cell. And of course, we need to give it a fancy name. And if the neuron is the, so let's do it this way. If the neuron is the presynaptic cell, then the target cell is the postsynaptic. So we have our postsynaptic cell, which is our target cell, whatever that target cell may be. And we know it has to have some specializations as well. So let's draw my target cell. The primary specialization it is going to need is some type of chemically gated channel. Right? We, for instance, talked about the chemically gated cation channels that's on our skeletal muscle cells. Right? Whatever it is, that chemical needs to be able to bind to it. And when it binds to it, it opens up that channel. But as we saw, for instance, on our skeletal muscle cell, there can be other specializations. For instance, like the invaginations, right? That increase the surface area of it or things along those lines. So there could be something else. But the key thing that we need on that postsynaptic cell is a channel that chemical, that neurotransmitter can bind to. And of course, as we also talked about, there is a third component. Let's undo that. Change the color one more time. There is a third component to our synapse, and that is the space in between. Because the key with a chemical synapse is that these cells don't touch, right? Electrical synapses, they touch. Again, think of the wiring in the wall. When they wired up your house, did they bring two wires really close together and hope that the electricity would arc from one end of one wire to the other end of the other wire? No, they took those two wires and twisted them together so they were hardwired together. They touch. Remember, our chemical synapse, they do not touch. There is a space in between. And of course, that space in between is the synaptic cleft. All right. So this is our chemical synapse. Really, if you think about it, there's no new information on the board. This is all stuff we've talked about. And so when we talk about this, there are some issues with a chemical synapse. It is going to be relatively slower than an electrical, right? Because after all, in electrical, sodium just goes straight from one cell to another. That's not what happens here. Here, our action potential comes down, the sodium comes down, it opens up our voltage-gated calcium channels, calcium comes in, causes the vesicles to bind to the plasma membrane, release the acetylcholine, which diffuses across the synaptic cleft and binds to the chemically gated channel, allowing sodium in this cell, and then sodium can continue down that way, or something like that. So it's a lot slower process because of this. The other big difference is, notice there's only one-way communication, right? Things just go from the presynaptic cell to the postsynaptic cell. With this, but the key that makes this so powerful, and the reason that this is the way our nervous system communicates, is that it allows for modification of the signal. Okay? When we can change how much neurotransmitters release, we can change how many receptors are here. So we can get bigger or smaller signals, right? The signals can be positive or negative. And so depending on the size of the signal, depending on whether it's positive or signal, depending on the number, we can get a number of signals. All of those things together sum to determine if the target cell fires an action potential. Because remember, most target cells are neurons, and what neurons care about is whether or not they're going to fire an action potential. And if we want to take it one more step forward, then if you think about it, the other thing is when that channel opens up, if you think about it, the action potential, let's change the colors of this to black. The action potential in this cell, in the first neuron, basically produces a graded potential in the target neuron. So the first neuron, oops, wow, that's bad. The first neuron's action potential produces a graded potential in the target neuron, and that graded potential, depending on the size, depending on the positive or negativity of it, will either may or may not, and let's say I put it that way, may fire an action potential. All right, and my typing skills continue to not get any better. Excellent. I saw there was a question. Electrical synapse is the all or nothing, while the chemical is not. No, I would not use that term all or nothing there, simply because when we talk about all or nothing, we're talking about action potentials, because action potentials don't change size. Now, the one thing I would say is if we had two neurons wired together and one fired an action potential, the other one would always fire an action potential. So I think the better way to think of an electrical synapse is that the cells are connected together, and so they work together as one. Whatever one does, the other one does. So it isn't necessarily all or nothing. One could just get a little bit more positive without firing action potential, but the first one gets a little bit positive, the second one's going to get a little bit positive. If the first one gets a little bit negative, the second one's going to get a little bit negative. They do the exact same things. So it isn't necessarily all or nothing because we're not talking about an action potential here. What we're talking about is that the cells work together as one, do the same thing. When you add two things together. So you are going to add signals together, and adding those signals together. So I mean sum from the math sense by addition, right? When you add things together, you get a sum. So if is the signal going to be big enough to fire an action potential? So the action potential is how a signal is sent from one end of a neuron to the other end of the neuron, but you are correct. How most neurons communicate with their target cells is by releasing a chemical signal through the synapse. That is correct. So the action potential is how the signal is spread down the axon, but the chemical that is released is how it actually communicates with the target cell. All right. Questions on that? All right. I made a pretty picture here on the board, but let's go ahead and erase it and take a look at the pretty words that say the exact same thing. So again, cells do not touch. There's that synaptic cleft in between. Our presynaptic cell, remember that is the neuron that is doing the communication, is going to release a chemical signal. And the generic term for that chemical signal is a neurotransmitter. It's a slow process because, like we said, we have to first open calcium channels, let calcium comes in, release the neurotransmitter via exocytosis, diffuse it across, bind it to a new channel, open that new channel, allow ions to flow through. This takes time. And it only allows one-way transmission of information. But the advantage of it is it's modifiable. It's not the same. They're not doing the same thing. A neurotransmitter release can both excite or inhibit that target cell, that postsynaptic cell, producing a graded potential that is either going to be excitatory or inhibitory. And that's really the key. These are two definitions, two terms we have to define. And I'm going to do it on the next screen, but I'll go ahead and do it here. Excitatory means the cell is more likely to fire an action potential. Notice the key word there. I didn't say that it guarantees that it fires an action potential. It's just more likely to fire an action potential. Whereas inhibitory means the cell is less likely to fire an action potential. But like I said, we're going to define those words in just a minute. We have a pretty chart that shows the same thing as well. So with variable signals, we may or may not get an action potential. Decisions are made and can be modified. Now, here is our synapse. Notice as you sit here and read through it, it is the identical process to what we did in our muscle cell. So I will quickly, rapidly go through it, but I'm not going to take the time to go through it again because A, we're behind on time, and B, you already know this. Action potential comes down the axon to the synaptic end bulb. That action potential is a big positive signal. That big positive signal brings the synaptic end bulb to threshold. And when we reach threshold, voltage-gated calcium channels open. When those voltage-gated calcium channels open, calcium rushes into the synaptic end bulb, and calcium triggers the movement of the vesicles containing the neurotransmitter to move to the plasma membrane and release their contents via exocytosis, releasing that neurotransmitter into the synaptic cleft. That neurotransmitter diffuses across the synaptic cleft to the target cell. On those target cells, there are chemically gated channels. Our neurotransmitter binds to that chemically gated channel, opens that chemically gated channel, and ions can flow into or out of our postsynaptic cell. And really, here's the only way we change the definition. Last time we just said it depolarizes the cell, and if the signal is bigger enough, it fires an action potential. But here, we can be more precise, right? Those ions flowing into or out of the postsynaptic cell are going to produce a graded potential in the target neuron. All right, that's really the only wording we have changed in any of this. But otherwise, it is the identical process to what we already identified, to what we already described. So I'm not going to spend any more time on that than that, because you had to know this for the last class. In fact, some of you had this as your essay question, right? Initiation of communication at the neuromuscular junction, boom, that's this process. All right, so I want to talk more about these graded potentials in that postsynaptic cell. All right, because like I said, that's what we care about. We care about how is this action potential in the target, in the main neuron going to affect the target neuron. As we talked about, binding of the neurotransmitter to the postsynaptic cell is going to open some type of ion channels. And ions are either going to flow into or out of the cell, producing that graded potential. And as we know, graded potentials, and I'll go ahead and write this here. Graded potentials can be depolarizing let's do it this way. Can be depolarizing or hyperpolarizing. And whether it's depolarizing or hyperpolarizing, remember, is determined by the ion channel that opens. So, what type of ion channel could we open that would cause a depolarization in our postsynaptic cell? Sodium, absolutely. Excellent. Sodium, because as we know, sodium is a positive ion that enters the cell, and that makes the cell more positive. Is there any other ions that we've talked about that technically could give you a positive? Well, but remember, potassium is a positive ion, but it's going to want to leave the cell. So if you think about it, you're right. Potassium is definitely one of the ions that will want to move, but remember, potassium wants to leave. And if a positive ion leaves the cell, that makes the cell more negative. Excellent. The other one that I was thinking of is calcium. However, we need to put an asterisk next to calcium, because remember, as we talked about, calcium makes cells do wonky things. So normally calcium isn't the positive ion we would want to come inside just to get a graded potential, because calcium runs the risk of doing something else. So technically, calcium could come in and make the cell more positive, but it's not the one that's normally going to be used. Normally, if we want to depolarize, we open sodium channels. To make the cell more negative, to hyperpolarize the cell, make the cell more negative, right? And that's, again, let's go ahead and add those into the definitions. Make cell more positive. We want to make the cell more negative. We can use potassium, but there is another common ion that isn't a cation. Instead, it's an anion that can be used to hyperpolarize. Anyone know what that anion? There you go, chloride. Chloride is a negative ion, and remember, it wants to enter the cell. So if we open a chloride channel, chloride's going to enter the cell, and that's going to make the cell more negative, because a negative ion entering the cells makes it more negative. And that's really the key. We are going to depolarize and hyperpolarize. Notice, once again, there's no new information on the board, but we are going to add one vocabulary term to it. Oh, no, I didn't mean to do that. I want that black. Right? So we do need to add one more vocabulary term. A graded potential that depolarizes, makes the cell more positive, is going to make it more likely to fire an action potential. And so we call that excitatory. It is an excitatory postsynaptic potential. Oops. It's an excitatory postsynaptic potential. And basically all that means is that it is a positive signal in the postsynaptic cell. And the key is it makes the cell more likely to fire an action potential. It doesn't guarantee it, but a depolarizing signal, and let's emphasize we are talking here about the depolarizing signal, makes it more likely to reach an action potential, to fire an action potential, more likely to reach threshold. So it's an excitatory postsynaptic potential. A graded potential that hyperpolarizes, therefore, makes it less likely to fire an action potential. And if it makes it less likely to fire an action potential, we call that an inhibitory synaptic potential. And these makes the cell less likely to fire an action potential. So, and again, because that's a big mouthful, once you've written them out once, you can abbreviate this as an EPSP, excitatory postsynaptic potential, and this as an IPSP, inhibitory postsynaptic potential. So again, really no new information here. We've just added a little to our vocabulary. And that's what we're going to see when I clear this. Right? These graded potentials are occurring in the postsynaptic cells, so we can call them postsynaptic potentials. And they can either be positive, where they're depolarizing or excitatory, or negative, where they're hyperpolarizing and they're inhibitory. And here we have the pretty picture that goes along with this. Here we have an excitatory postsynaptic potential, Notice again, they've put their threshold at 55. Different books put it at different locations. We're going with 60. We're sticking with 60. But the point is, notice it's getting us closer to threshold, but it doesn't actually have to get us there. It makes it more likely to reach threshold, but it does not have to get us there. Even if it just gets us 1 millivolt more positive, it's still excitatory. That's not going to get us to threshold by itself, but it's still considered excitatory. And like I said, typically these are going to involve our sodium channels. Inhibitory makes the cell more negative, moves it further away from the threshold, causes that hyperpolarization, makes it less likely to reach threshold. And for that, we typically use either chloride or potassium channels. All right. Questions on that? And here's the deal. Notice I'm using terms like more likely and less likely because remember when we first talked about these things, when we talked about our skeletal muscle was very special. Remember with our skeletal muscle, we talked about how 1 neural action potential is going to produce 1 muscle action potential. There's a 1-to-1 ratio. In the nervous system, it's not a 1-to-1 ratio. And that's really the last key piece of information we need to add to this to really understand. In the nervous system, 1 neural action potential in 1 neuron does not produce a neural action potential in a second cell. So 1 excitatory postsynaptic potential will not generate an action potential. Because remember, we don't want them hardwired together. Our skeletal muscle, we want hardwired. We send a signal to our skeletal muscle. We don't want our skeletal muscle to think about what it's going to do. We want it to contract. But in our nervous system, we want to be able to think about it. We want to be able to make decisions. Heck, this is something, if you think about it, we have already talked about. Because if you think about it, our resting membrane potential is negative 70 millivolts. Our threshold is negative 60 millivolts. And therefore, oops, too many M's. So if you think about it, the difference is, a 10 millivolt change. If you also remember, the first time we talked about graded potentials, we said that on average, right, we said they were small. And the number we threw out there was somewhere, plus or minus 2 millivolts. So if you think about it, 1 graded potential, which is just positive 2 millivolts, isn't going to get a cell that's at negative 70 to negative 60. So while we may not have emphasized it at the time, we already hinted that this was going to be the case. Now, luckily, a neuron does not have to rely on just 1 release of chemicals from 1 action potential. A single neuron, on average, in the central nervous system, so in our brain and spinal cord, a typical neuron will have something between 1,000 and 10,000 synapses. So 1 neuron can be receiving 10,000 different graded potentials. And that's what it's adding together. And here's the great thing about them, right? Basically, and I've said this term before, right? Neurons are bean counters, right? They are essentially, I'll put the bean counters part in parentheses, right? They are essentially accountants, right? Any good accountant is capable of balancing the checkbook, right? That's what they do. They balance a checkbook, right? They count the money going out. They count the money coming in, right? Money coming out is obviously negative. Money coming in is positive. and then they add it together, right? They sum it. And depending on the outcome, right? Depends on whether or not you have a good month or not, right? If there is more positive than more negative, oops. If the positive coming in is greater than the negative going out, then everybody's happy, all right? You can make $1 million a day. But if you have $1,001 going out in salary and expenses, are you going to be happy making $1 million a day? No, absolutely not, right? Because the money going out is greater than the money coming in. Our neuron has 10,000 different inputs coming to it. So let's use some really simple numbers. Let's say that 9,000 of those are excitatory postsynaptic potentials, and 1,000 of them are inhibitory postsynaptic potentials. Do you think this cell is likely to fire an action potential as a result of that? I'll tell you right now, excitatory and inhibitory postsynaptic potentials cancel each other out. So 1,000 of these cancel out 1,000 of those, but we still have 8,000 excitatory postsynaptic potentials. Yeah, so this will almost certainly fire an action potential. Conversely, if we have 5,000 excitatory postsynaptic potentials and 5,000 inhibitory postsynaptic potentials, this cell is receiving a tremendous amount of information. But is this cell going to fire an action potential? Yeah. No action potential. It's getting 10,000 pieces of information, and the net result of those 10,000 pieces of information is it does nothing. It didn't get enough of a positive signal to get it to reach threshold. Now, if a single excitatory postsynaptic potential was big enough to reach threshold, then yes, technically one excitatory postsynaptic potential could do that. Let me sneak in a quick drawing. Remember, we have our cell body, we have our axon, and our goal, and we'll do this in red, remember, is to get the signal here to the axon hillock. We know that our cell body has dendrites. Let's just draw one dendrite just to make this, no, hold on. Undo that, let me go back to blue to keep this consistent. We'll just draw one dendrite here, again, just to make this super simple. All right, so let's think about this, and we'll do this in my highlighter. And we'll use green. So right here, I have a synapse from another neuron that communicates with it. Heck, we'll even draw it. Cell body, axon, synaptic end bulb, releases its neurotransmitter. And right here, we produce a graded potential. I'm just going to GP just to keep it small. All right. So right here, we produce that graded potential, or more specifically, we'll be more specific, an excitatory postsynaptic potential. We produce that excitatory postsynaptic potential on this neuron right here. That excitatory postsynaptic potential has to spread down the dendrite, spread down the axon cell body, the neuron cell body, to get to the axon hillock. And remember, these graded potentials spread passively. And remember, as things spread passively, they attenuate. They get smaller as they travel. So theoretically, if you produce a big enough signal here that by the time it spreads to the axon hillock, you still get a positive 10 millivolt change, then yes, absolutely, you will reach threshold and you will fire an action potential. But for that graded potential to spread that far, getting smaller as it travels, this would have to be a huge signal that we produced here at this synapse. And remember, graded potentials, excitatory postsynaptic potentials, are typically not big. So it might need to be a 40 millivolt change, right, or something like that, to get that change to spread passively to the axon hillock and reach threshold. Can it happen? Possibly, especially if you think of it this way. If, for instance, the synapse connected not to a dendrite, but if the synapse connected to the cell body, like right here, and all we had to travel was a super short distance to get to the axon hillock, it might be possible for one excitatory postsynaptic potential to get it to fire an action potential. But at that point, why not just hardwire it together? So yes, mathematically, if it was big enough to get it to reach threshold, it would reach threshold and it would fire an action potential. But usually these waves are too small to be able to do that. Yes, and this is one neuron communicating with another, right? Or in this case, now I've got two. One neuron communicating with a second and a third communicating to that second one as well. Because remember, most target cells, as we talked about, are neurons. Most neurons talk to other neurons. So when we talk to other neurons, we want to get them to fire an action potential. Awesome questions. Any others? All right, I've written it out here. We've done our checkbook balancing, and that's really what this is. Yes, so eventually that motor neuron will go out and talk to the muscle cell and get the muscle cell to contract. All right, so again, you think about it. In that pathway that we've already talked about, if you think back to when we were talking about contracting a skeletal muscle, we talked about making a decision and sending that signal down to the muscle. It actually takes 2, 3 neurons to do that. One neuron to go from the brain, one to cross, one to go down the spinal cord and then go out. So yeah, so typically, eventually you'll lead to other targets. They can lead to other targets, but most of the target cells are going to be neurons. So again, it's bean counting. One inhibitory postsynaptic potential cancels out another one, and the goal is to add them up. And there are 2 ways to add them up. I know this may be a little confusing, but your book also has this really, and when I first read it, I thought it was incredibly silly. But the more I think about it, the more I really love this analogy that your textbook uses. Your textbook uses this idea of reaching threshold, right? Our goal is to reach threshold. It's a little too small now. And we want to reach threshold with these little waves by adding these small waves together. And the analogy your textbook uses for this is to fill a bathtub. So what I have here is a bathtub. And my goal to fill it is to use a bucket. I have a bucket, and I need to use this bucket to fill the bathtub. And as it turns out, it takes 10 buckets of water to fill the tub. Right? Filling the tub is reaching threshold. Right? So if you think of one bucket as being a 1 millivolt change, and you have to get from -70 to -60, then you need a 10 millivolt change. You need 10 buckets to reach threshold. All right? But here's the problem with your bathtub. The problem with your bathtub is your bathtub has the drain, and you don't have the stopper for the drain. So let's think about this. Your bathtub is on the 2nd floor, and your faucet of hot water is in the basement. So you have to go to the basement, fill your bucket, walk all the way up to the 2nd floor, dump your bucket into the bathtub, go back down to the basement, fill the bucket, and come back up. By the time you come back up with that bucket, the second time, what's happened to the first bucket of water? By the time you walked all the way down to the well, if it takes you, then you're walking fast. Because for me, walking down all the way to the basement, 2 flights of stairs, filling that bucket up with a faucet, bringing it all the way back up 2 flights of stairs. By the time I get back up, that first bucket is not just half gone, it's all gone. All right? So I can't fill it that way. I need 10 buckets of water in here, but I have to get this water in here fast, right? Because it leaks away. To fill the tub, I have to get many buckets into the tub, and I have to do it fast. Do it quickly. All right? So notice instead, if the faucet wasn't in the basement, but was in the right next to it, then I could fill the bucket and dump and fill the bucket and dump and fill the bucket and dump and fill the bucket and dump. And if I did that 10 times really quickly, then I could fill this before it all drains away. Does that make sense? So one way I can do this, one person by themselves adds 10 buckets really quickly. All right, that's one way we can do it. Another way is I can have me and 9 of my closest friends. So 10 people all dump 1 bucket of water at the same time, or in the same place, like in the same place. All right. If me and 9 of my closest friends all stand around that bathtub and all dump our buckets in at the same time, we can fill the tub up. So notice, to fill the tub to reach threshold, there are 2 ways we can do it. One person can do it by themselves by doing it really quickly, or many people can do it all by doing it at the same place. If 10 people all take 10 buckets and all dump them in the same place, we can fill it that way too. Does that part make sense? I think that's an old yes. Okay, that's a new yes, definitely. Excellent, perfect. Those are your new yeses, perfectly. Excellent. So all we have to do is give these ways special names. All right, so if it's one person, or in this case, one neuron, who produces 10 action potentials really close in time, because they're occurring close in time, we call this temporal summation. This one, where I have 10 people, all in the same people, all dumping at the same time. True, they do need to all be quick. The 10 people do need to dump it at the same time, but more importantly, they have to do it in the same location. As long as they're in the same location and doing it, then yes, it all has to be around the same time. So you're right, time is an issue there as well. But because they're all doing it at the same location, 10 different people at the 10 location. Because if I have 10 people who all simultaneously dump their buckets, but one's here and one's in Seattle, Washington, and one's in, Reno, Nevada, and another's in Cincinnati, Ohio, and all different locations, the fact that they're doing it at the same time doesn't matter. What matters is they do it at the same location. And if they all do it at the same location, we call that spatial summation. And so that's really the take-home message. Neurons are going to use temporal and spatial summation to add those thousands and 10,000s of graded potentials together, those excitatory and inhibitory postsynaptic potentials together, and determine whether they fire an action potential. I can repeat it, and I've got a pretty picture that goes along with it. So let me go ahead and clear this. Clear all drawings. So like I said, 2 ways to add up, spatial and temporal. So notice here, we have our temporal summation. Temporal summation, remember, temporal summation It is one synapse that fires multiple times, close in time. And if they fire multiple times close in time, they can add the waves together to reach threshold. One stimulus by itself doesn't reach threshold. But if it immediately fires another one, we can add those waves together. And as we add those waves together, we get an action potential. Conversely, with spatial summation, we have, in this case, multiple neurons all fire action potentials. And when they all fire action potentials together, they are able to add up and reach threshold and fire an action potential. And so it is by adding many signals together. Both, we use both. All neurons use both, right? Because again, if you think about it, these two neurons, this could fire 5 times, this could fire 7 times. And there could be other ones as well, right? It all gets more complicated. We could have a third one coming in here, but this one could be sending negative signals. So this is sending negative signals, and this one's sending positive signals, and this one's sending positive signals. You add all those signals up and determine whether or not you reach threshold. I think someone just raised their hand. Hold on one second. I closed my chat window. Let me bring that up. There it is. Oh, there it is. All right, there was a question? Someone raised their hand? I thought I saw
Yeah.
someone. Yeah,
no,
okay. Yeah.
I had a question. So basically, you said that the temporal summation is one neuron firing rapidly.
Mm-hmm.
And you said that the spatial summation is multiple neurons all firing at the same time in the same place.
Correct.
Okay.
And so, yes, absolutely, as it says, both of these are going to be used. Remember, as we said, a single neuron typically gets between 1,000 and 10,000 synapses. And each of those neurons talking to it can produce action potentials at a rate of 100 action potentials per second or something crazy like that, or at least 10 per second or something like that. So there's a massive ability to be able to produce lots of signals, lots of positives, lots of negatives, and they all add up. We count all those beans and determine whether it fires an action potential. All right. All right, excellent. The last little bit, and again, not too much new, but the last little bit we need to talk about is just some definitions. As I mentioned, we've mentioned a couple times, there are many, many neurotransmitters. Over 100 different neurotransmitters have been identified in the human body, and that doesn't even include what's going on in other species and animals. They can be excitatory or inhibitory depending on the postsynaptic channel, right? Probably the best example of this, an important example of this, is acetylcholine. Let's draw 2 neurons. Here are 2 neurons. Both of these neurons release acetylcholine as their neurotransmitter. But that doesn't mean that acetylcholine is going to have the same effect on the target cells. Let's draw some target cells. One target cell can be our friend that we already know about, skeletal muscle. What effect does acetylcholine have on skeletal muscle? Is it excitatory or inhibitory? Excitatory, absolutely. Right? It causes it to contract. Right? It stimulates it. It's excitatory. We already know that. Excellent. But it turns out, and here's what's really interesting. You can take that exact same acetylcholine. No. And you can take that exact same acetylcholine and instead spread it onto cardiac muscle. And on cardiac muscle, it turns out that acetylcholine is actually inhibitory. It slows the heart rate down. On the surface, that doesn't seem like it would make a lot of sense. This is the same key, and if the same key unlocks the same door, then it should have the same effect. But that's the key to this process. It's not the same door. On our skeletal muscle, the door that acetylcholine opens is a cation channel, as we know. But as we also know, it is primarily sodium that passes through it. And so as we know, that sodium, which is a positive ion, is going to enter into the cell through that channel, and it makes the cell excitatory. Well, in our cardiac muscle, it turns out what it opens is a completely different type of channel. On our cardiac muscle, it actually opens a chloride channel. And so, of course, when you open a chloride channel, that negative chloride ion is going to enter into the cell, and it inhibits the cell. It slows the heart rate down. So notice, whether it's excitatory or inhibitory doesn't depend on the ion, it depends on the channel. Yep, so it would hyperpolarize it, exactly. So in this case, it slows the heart rate down, it hyperpolarizes the cardiac muscle, and the cardiac muscle contracts slower. The heart rate slows down. Whereas this one depolarizes, exciting it, and it fires an action potential and it contracts. So notice, whether a neurotransmitter is excitatory or inhibitory depends not on the chemical, but on the channel that it opens. All right? So if we clear that, then we can emphasize this. Oh, I think you know what has the excitatory effect on the heart. Anyone know what the neurotransmitter is, or the hormone that is released into the blood that increases your heart rate? Not endorphins, although that's close. Adrenaline, or what is also known as epinephrine. Epinephrine and norepinephrine, which are the fancy terms for adrenaline and noradrenaline, right? Which are the exact same things as adrenaline and noradrenaline. Oops, not all one word. Oh, and it's not a G. So adrenaline and noradrenaline, or epinephrine and norepinephrine, which is also spelled wrong. Those are the ones, well, to slow the heart rate down, right? After you're done exercising, you know, if you get up and run around the room 15 times, or let's say you're growing chickens, and once you release those chickens in the backyard, you have to chase them down. Chasing down chickens, excellent exercise. During that time, you might want to increase your heart rate so that you have more blood and more oxygen and more nutrients going to your muscles. But once you're done chasing those chickens around, don't you want your heart rate to come back down? Or maybe if instead, after spending 45 minutes watching the news, right, you're totally stressed out. So what do? You go down in your lotus pose, you light a couple candles, you close your eyes, and you just sit there and go, and as you sit there meditating, what happens to your heart rate? It goes down. How does it go down? because your parasympathetic nervous system is releasing acetylcholine to open up chloride channels to slow your heart rate down. Yep, adrenaline and noradrenaline is its own neurotransmitter that opens up those channels, that opens up sodium channels on our heart cell and makes it beat stronger and faster. Noradrenaline and adrenaline would be fine to use on the test, and as opposed to epinephrine and norepinephrine. That is correct. Both are completely acceptable terms. And like I said, things that we'll talk about in a lot more depth when we get to the autonomic nervous system, which is what affects the heart, obviously. So yeah, we'll definitely get to all of that before the next test in the next couple weeks. Wow, we only have a couple weeks left. That's crazy. All righty, so much material to cover. You guys are so screwed. All righty, excellent. Again, one of the advantages of this system is it's not hardwired. There's flexibility in the system. One of the ways is, again, we can fire more action potentials. That will release more neurotransmitter. But think of it also, we could also make more neurotransmitter. If we pack more neurotransmitter into a vesicle, if we put 20 acetylcholine instead of 10 in a vesicle, when that vesicle binds and releases, it's going to release more neurotransmitter and you're going to get a bigger signal. So not just how much you release or how much you make, also how long it's in the synaptic cleft. Some of you had the question of what happens if you remove acetylcholine esterase from the synaptic cleft. And as some of you got correct on that, that slows the removal of acetylcholine. Acetylcholine still goes away eventually. It can still diffuse out. It can still be endocytosed, but it stays in the synapse longer. Those invaginations of the motor end plate keeps the neurotransmitter in the synaptic cleft longer, right? Gives you more surface area. So these are all ways that we can modify it and increase the number of postsynaptic receptors. These are all ways we can change how strong a connection is between two cells. I wouldn't say necessarily they would contract for longer because, again, remember, we're outside of muscles now. We are now into neurons. But what would happen is it would stimulate the cell for a longer period of time. It would produce more graded potentials, excitatory graded potentials. And those excitatory graded potentials can either get a skeletal muscle to contract, can get a gland to release its secretion, can get the heart muscle to contract more, or can make it more likely that a neuron fires an action potential. So it's more stimulation, more excitation to that target cell, which means that the target cell is more likely to do whatever that target cell is supposed to do. And then we even also talked about blocking or activating the neurotransmitter at the receptor site. And we talked about things like curare or tetanus, where we're blocking and we're activating. So those are other ways we can modify chemical synapses, and we've seen that as well. Last thing I need to do is just identify some of your neurotransmitters. Your book's got a huge list of them that you can look at if you like. I just want to highlight a few of them. Acetylcholine, remember, as I talked about, is the most common in the peripheral nervous system. And as we talked about, it can be both excitatory and inhibitory, depending on its target and the receptors. Some neurotransmitters are amino acid based, glutamate and GABA, and I know I don't normally allow you to have abbreviations, but gamma aminobutyric acid is incredibly commonly known as GABA, and I will actually let you use the abbreviation GABA on this exam. Glutamate is the most common excitatory, and GABA is the most common inhibitory neurotransmitter. So these are the most common in the central nervous system. Some hormones can be molecularly more complex. We talked about the examples of hormones like norepinephrine and epinephrine. Those are our stress hormones. Dopamine, right, as we're learning more and more about dopamine, plays an important role in social interactions, in pleasure sensations, things along those lines. Speaking of pleasure sensations, endorphins, right? This is the type of hormones that play a role in that love and chocolate, Endorphins play an important role, or endorphin stimulators are things that play an important role in addiction. Things that we become addicted to are more likely to increase endorphin release. Substance P is a really interesting neurotransmitter that seems to play an important role in our pain pathways. That's why it's called Substance P, because it's related to pain. Our pain pathway is one of our least understood pathways. We do know many of them, as we talked about, are unmyelinated, but there's a lot of complexity to it and a lot of research going into that. So Substance P is one of those really interesting. And then there's some really unique things that can be neurotransmitters. ATP can actually be a neurotransmitter. Some gases, like nitric oxide. I don't mean nitrous oxide. I know you're all locked up at home, but back in ancient times on Friday night, you used to be able to go to the rave and get a nice big balloon full of nitrous oxide so that you could have a good time while you're partying with your glow sticks. Or maybe you're more legit, you just had to go and get your tooth extracted. So you went to the dentist office and used some nitrous oxide for that as well. Well, nitric oxide is similar to that, and it also has a similar effect. Basically, it dilates smooth muscle. So, like for instance, when you dilate blood vessels to bring blood to the surface when we exercise, or to increase blood flow to the kidneys or things along those lines, we can use nitric oxide to change the diameter of our blood vessels to move blood to different parts of our body. And like I said, your book's got a nice table that talks about all the different types, so you can see some of the other ones. But these are the common ones that we've talked about, or we'll talk about as we move in from here into the rest of 430, and even into 431 as well.
Professor,
yes.
Did you say that glutamate was excitatory?
Yes, glutamate is the most common excitatory neurotransmitter in the central nervous system.
Okay.
And GABA is the most common inhibitory in the central nervous system.
And the other ones are peripheral?
Well, acetylcholine, remember, is the most common. So epinephrine and norepinephrine, many of the rest of these that are listed here are used in both the central and the peripheral nervous system.
Okay.
So they can be used in multiple locations. And notice I didn't necessarily say excitatory inhibitory, because remember, they can have complex interactions depending on what the receptors are. But these first three are the most common ones, so it was worthwhile to mention that. For the peripheral, acetylcholine is the most common, both excitatory and the most common inhibitory. And glutamate and GABA are the most common in the central nervous system. But as I said, there are over 100 different neurotransmitters that have been identified. All right. So with that, we are done with the physiology of the neurons. So what we can do now, finally, when we come back from the break, is start to focus more on the anatomy, which is one of those things that we've been wanting to talk about for a while. So let's go ahead and take our first break now. We will take a 10-minute break, and as I look at my phone, I see that it is 1:23, so we will return. So restart at 1:33, and I will start recording at that point. All right. Well, I know I spelled it wrong, but I'll know what it means. All right. Any other questions before we take our break? All right. I will go ahead and pause our recording. All right. So what we have here, as we talked about, and we talked about this a little bit in the last class. We also talked about this some during the histology lecture as well, but this part we talked about in the last class. And this is where we have our 5 ventricles, as we talked about, those cephalons. That we are going to use to identify and distinguish the different parts of the brain. They start early, 6 weeks of development, but as we also talked about, they form precise structures that can be seen and identified in the neural anatomy. All right, here we see an intact brain, and they've shaded the internal structures to show us where they are. And then here we see a sagittal section through the brain where we can see some of these structures as well. Notice As we look at this, we see some of the things that we've talked about. Down here, we have our cerebellum. And remember, as we talked about for the cerebellum, we have the white matter. That is the central part of our cerebellum. And then the outer part, which is the gray matter. And the gray matter, as we talked about, form those ridges, those folia, and then the indentations are the sulcus. So we have those sulci and those folia that form the organization of our cerebellum. And remember, now here you can see the white matter forms this elaborate branch-like structure that, again, we call the arbor vitae. So those are structures that we talked about, and they're also on your list and things we'll discuss as well. All of this up here is the cerebrum, and the cerebrum is the entire structure that is formed by the telencephalon. So essentially, telencephalon and cerebrum are essentially identical terms. As I believe, I have a picture, a pretty picture that shows, perfect. Here with a coronal section through the brain, we can actually see that that cerebrum is divided into two hemispheres. On those two hemispheres, remember, we have those invaginations like we talked about. So again, an indentation is a sulcus. And that is the same as the cerebrum. However, the raised ridges, remember, we called a gyrus, or the plural being gyri. However, if you will notice, there are some sulci that are very deep. And a very deep sulcus, like this one right here on the midline, is called a fissure. And these are generic terms. So there are many sulci, many gyri, and even a couple fissures. However, some of these structures have specific names. This particular fissure that is located in between the two hemispheres, right on the midline of the cerebrum, is what is known as the longitudinal fissure. And it is called the longitudinal fissure, as we see from this illustration here. The longitudinal fissure runs the entire length of the cerebrum, separating the brain into those two hemispheres. So as you look at the list, as we look at some of these things, we see gyrus, we see sulcus. Again, these aren't, this is a gyrus, and that's a gyrus, and this is a gyrus, and this is a gyrus, and this is a gyrus. This is a sulcus, and this is a sulcus, and this is a sulcus, and this is a sulcus. So really, they're just structures, right? All of these things that I've talked about so far, a gyrus, a sulcus, even a fissure, these are all things that are structures. But some of these structures do have specific names, like the longitudinal fissure. Like, notice if we go back to this brain picture, right from the top of the brain, there is a sulcus that starts at the top and works its way down. And notice, even though they've put the term fissure here, this is not a fissure. This is actually called, and let's see, I think I have a pretty picture that shows it. It is called the central sulcus. This is it from the top. But where I have an even better picture of it is right here. Here, notice they've color-coded it for us. So this structure right here that starts right at the top, and this invagination that goes down is the central sulcus. Notice there is a gyrus in front that they've highlighted in red, and that is the precentral gyrus. There is a gyrus posterior to it, the central sulcus, that they've highlighted in dark blue, and they call that the postcentral gyrus. So these are all structures that we've identified so far. The other thing that this picture illustrates nicely is that the cerebrum is divided into 4.5 lobes. And they've color-coded them here for us. These lobes happen to be, as you can see, identified by the bones associated with them. So we learned those cranial bones that form the cranial cavity. We've learned them once, we get to use them twice. Your forehead is the frontal bone, and it covers the frontal lobe. The bone on the back of your head is the occipital bone, and it covers the occipital lobe. Your temporal bone forms the lateral inferior part of your skull, like where your ear hole is, and that is where your temporal lobe is. And superior to that, on the sides, lateral but superior to the temporal, is our parietal lobe. Great question. No, they are not general regions. These are actual precise anatomical structures. Notice, for instance, that central structure, that central sulcus divides the frontal lobe and the parietal lobe. Notice there is another sulcus. Remember when I keep drawing the brain, I keep talking about having this mitten or the cerebrum. It looks like this mitten-like appearance where you have the thumb and then you have the rest of it. Notice this is the thumb, this is the rest of it. And notice this line right here. Let me undo that. Use the highlighter because it's easier to see. This, and I don't like that color. We'll go ahead and use the brown. This sulcus right here is the lateral sulcus. And notice that lateral sulcus separates the frontal lobe from the parietal lobe. Notice here for the occipital, they've kind of got just this kind of what appears to be somewhat random looking line that is drawn there where it might be more general region, but it turns out there are anatomical structures that identify the occipital lobe. They're just deep from the surface. They can't be seen from the surface like this, and they're not structures you're responsible for. But the lateral sulcus and the central sulcus are structures that are on your list. The precentral gyrus and the postcentral gyrus are structures that you are responsible for that are on your list. These are all structures that we have talked about so far. And of course, the lobes are structures as well. All right? With me so far? The postcentral gyrus does kind of look a little general. I do agree with that. The key to that, and again, part of this you have to remember is the bit of artistic license that our artist has made for this. But what you can clearly see, and we'll see this on many of the pictures, is the central sulcus is always a fairly obvious structure. It starts at the top of the head, it projects anteriorly, and it goes towards, but notice it does not touch the lateral sulcus. So all of this gyrus right here in front of it is the precentral. All of this gyrus behind it is the postcentral. So really the key is to find the central sulcus, and then the ridge in front of it and the ridge behind it are going to be your pre and postcentral gyri. Notice if we go to some of the other pictures we've looked at, here is our, and let's go ahead and draw it. Starts at the top of the head and projects down. Starts at the top of the head and projects down. So that is the central sulcus. Here in red would be the precentral gyrus. Here in blue would be the postcentral gyrus. No, the difference between a sulcus and a fissure is really the depth. So, and even then, again, as we know, anatomists hate us, so it's not quite clear. So notice when we go back to that coronal section, here we have an indentation that is super deep. That is a fissure. Notice here is that lateral sulcus. Notice that lateral sulcus that we talked about is also very deep. Right? When you compare this sulcus to the depth of this sulcus, the lateral sulcus is very deep and so should probably called a fissure, but it's not. It's called a sulcus. If you said greater tubercle instead of greater trochanter for the femur, did you get that wrong? Then yes, you would get it wrong if you put longitudinal sulcus instead of longitudinal fissure. You would lose partial credit for that, yes. Names matter. Yep, welcome to Anatomy and Physiology. All right, so we have our gray matter. And again, notice like the cerebrum, our gray matter is superficial, our white matter is deep. And here on the cerebrum, and this is important, the gray matter of the cerebrum, right, as we see here, most is superficial, and it is called the cortex. So notice I'll grab my highlighter again, and in blue, all of this that I'm drawing on the surfaces is the gray matter of the cerebrum. And like I said, the gray matter, I'm not even going to finish. The gray matter of the cerebrum is the cerebral cortex. All right, so this is our cerebral cortex. But notice also there's that key word most. And notice down here, there's this brown and this pink and this pink and this purple and this red. There are some other structures in here. Notice these are surrounded by white matter. Remember, as we talked about, the white matter of our cerebrum, or really the white matter of the central nervous system, we call tracts. So here we have some clusters of cell bodies that are surrounded by the white matter, right? They're deep in the white matter, and those we call, remember, nuclei, right? Nucleus singular, nuclei is the plural. So here I have some nuclei and cortex and white matter tracts. So here we see the cortex and the nuclei. That is the gray matter. And here we see some examples of the tracts. Notice some tracts are local. If you think about it, when you're feeling something with your hand, you need to know what your thumb and your index finger are feeling. So those might be related to each other. Some are going to be longitudinal tracts, because maybe what you're thinking about and what you're seeing and what you're hearing need to be connected to each other. But also, at the end of lecture, when you guys all stand up and applaud me, that ability to put your hands together coordinates what's going on from your left and your right. And for those, we need what are referred to as commissural tracts. A commissural tract is one that goes from one hemisphere to the other, coordinating what happens in our two hemispheres. So we have short, long, and commissural tracts of white matter that can be found as well. So notice when we look at, and again, these so far are the telencephalic features. These are just characteristics that we've been identifying. Again, the lobes, frontal, parietal, temporal, occipital, right? Fissures, gyri, sulci, all the things that go along with that we've talked about. And while we have many, many great models in the classroom, one of the reasons why this exam is typically a PowerPoint presentation is because of illustrations like this. As you get more comfortable and more familiar, you may be able to, like I am, to quickly and readily find that central sulcus and identify it, which allows you to see the precentral gyrus and knows where the frontal lobe is and so on and so forth. But it can be tricky. However, if we have nice, pretty color-coded pictures like this, then it's much easier for you to be able to identify the central sulcus or the precentral or postcentral gyri or the lobes and things along that. There's one other thing about these illustrations. So again, you can do it here, but it's trickier. The fact that it has labels makes it nice, but it can be a lot trickier on the models or things like that. There's one other reason I like using the illustrations, and that's what's illustrated here. Notice there are these numbers that are on this brain. These numbers are what are known as Brodmann numbers. Because back in the late 1800s, early 1900s, there was an anatomist by the name of Corbinian Brodmann. Now, again, late 1800s, early 1900s, we didn't have the microscopes or the other types of materials and things that we have these days for studying the brain. So what he did, I should say, not what I did, he started with a cat brain. And what he did, it started with that cat brain, is he looked at the cat brain, one cat brain, and looked at its characteristics. And then a second, and a third, and a fourth, and a fifth, and a sixth, and a seventh, and an eighth, and a 147th, up 1D. And when he looked at all these hundreds of cat brains, what he noticed is, every single one of these cat brains that I have here has this kind of raised ridge right here on the center. And it just happened to be the very first thing that he recognized that all of these brains, while there were some variations, all of these brains had in common. And so guess what he called it? Well, I highlighted it for you. He called it Area 1. And then he found a second feature that all of them had in common. And he called them Area 2. And then a third, and then a 14th, and then a 45th, and so on and so forth. And then after doing this with hundreds of cat brains, he did the same thing with monkey brains. And then he did the same thing with human brains. And he found these same consistent structures. These Brodmann numbers represent structures, right? That's it. Did he have any idea if those particular structures had a specific function or what that function was? Absolutely not. But it was something that he found that was consistent. At least the anatomy of these brains, not just within the species, but across species, were fairly consistent. And as it turned out, as our understanding of neuroscience grew, what we found is that there is actual remarkable similarity. It's not identical, but many of the consistent structures he found in the brain actually correspond to specific functions. So many of these structures correspond to specific functions. So, and I will both answer the question and finish my thought on this. You do not have to memorize the Brodmann numbers. On the exam, I'm not going to ask you, what is the function of Brodmann's Area 17? However, as it turns out, Brodmann's Area 17 is a structure of the brain that, as it turns out, has a specific function. And that specific function, as you can see, is the primary visual cortex. This is where your brain perceives light. So the same way having numbers on the arm and leg models helps us to be able to recognize things as we're looking at it, I can say, hey guys, Area 4 is your primary motor cortex, and everybody knows exactly what I'm talking about. So I will use pictures like this. I will use pictures like this during the exam because it will help you. Because again, as I've said before, I want you to show me you have the information. And so the more obvious I can make the pictures, the easier that's going to be. That doesn't mean you shouldn't study the pictures of the models and the pictures of the charts, because I guarantee those will be on the exam, especially when we get to the cranial nerves. When we get to the cranial nerves, there are some great pictures of the cranial nerves that are available. I'll make sure that they are on that CRC site, and if they're not, I'll add them to our site so that you can study them, because I guarantee they'll be on the exam as always. But for a lot of the cerebrum, there is an advantage to being able to use pictures like this. All right. I know that seems like a broad introduction, but at the same time, we've also been talking a lot about the basic anatomy, the basic structures you are responsible for. All right. So what that leaves us with now is to start talking about, notice for each of our lobes, and maybe this is a picture, I like this picture better. Each of our 4.5 lobes have different regions in them that have different functions. as I mentioned, one of the big differences between nervous tissue and any other tissue we've talked about is that our nervous tissue has specialization and it has lateralization. Right? As we talked about, the rectus femoris and the bicep femoris basically do the same thing. They're muscles that contract. And if you cut a little piece of one of the muscles out and cut another piece of another muscle out and switch them in their locations and assume you could do that without damaging the tissue, it would work identically. The same is not true for your brain, right? What this part of your brain does and what this part of your brain does is something entirely different. And even if this sulcus and this sulcus are similar in shape, you couldn't take them and replace them and they wouldn't function the same way. And I'm not just saying that because obviously we know nervous tissue doesn't regenerate. I'm not talking about it being damaged. I'm talking about it does different things, right? So the different parts of the brain do different things. Not only is this, as we said back here, going to be vision, whereas something here up here might be our movement. But what is also interesting is that as we go back to this picture, what this, so for instance, and let's just go some quick squares. This part of the brain is movement of your right arm. This part of the brain in the same location on the other side is movement of the left arm. So arm here, right? That is clearly different from head here, or face here, face here. So there's specialization. This does head, this does face, or this does hand, this does face, this does hand, this does face. They have different regions to it. But what's also interesting is that what this area does on this side and what this area does on this side, even though they're the same locations, could be different. So it's not just left and right. One of these may be speech and the other may be your spatial awareness of where you are in space. And that's lateralization. Lateralization is that what happens on one side is different from what happens on the other side. And specialization is what happens in one area is different from what happens in another area. So yes, no. So to answer the question, so here's the list. You are responsible for memorizing the functions of the areas or regions, and that's really, think of it. I will use the term area or use the term region. And what that is shorthand for is the area or region of gray matter, of cerebral cortex, right? So when I say area or region, what I'm talking about is this is the cerebral cortex, the cells. So again, if we go back to this picture here, these cells here have a function, and these cells here have a function, and these cells here have a function. So when we look at it from the top, and I say this area here is area X, and area X does Y, what we're talking about is that little bit of gray matter that has a name as an area and has a specific function. Now, all 45 of Brodmann's areas don't necessarily, and again, 47, there's more. I think he had like 80 or 100 or something like that, different areas that he identified. You don't need to know the function of every single one of these numbers. What you do need to know is the function of every single area that is on your list. So as you look for your frontal lobe, how many frontal lobe structures do you need to know? How many, let me say that again. How many frontal lobe regions are you responsible for identifying?
3.
3, excellent. So you need to be able, so as I point to those 3 things, what is this, what is this, what is this, not only do you have to be able to tell me what that area is, but you also have to tell me what the function of that area is. Okay? With me so far? I know we haven't really talked about anything yet, so let's start. Excellent. So as we look, the first area, and I think I will stick with the highlighter because I think that's useful. There's my highlighter, and let's stick with black to make it stand out. What we are going to talk about first is the frontal lobe. So the frontal lobe is all this part here that is in pink and red. The first part, as you can see, because it's been emphasized here, is the prefrontal cortex. And the prefrontal cortex is basically, as the name would indicate, the very front of your frontal lobe. So the very front of your frontal lobe is the prefrontal cortex. That is an area you are going to be responsible for identifying. And as we talked about, you need to know the functions of these areas. Now, here's the fun thing about the study of neuroscience. How do we know what these areas do? Anyone hazard a guess?
We innervated them and tested them to see what triggers it.
True, and that is something now, sure, scans, but we've been able to only do scans, really good scans, for probably about the past 20 years, really good scans for about the past 5. We've been able to scan the brain non-invasively for 15 years, something like that. What did we do before that? Did we just have no idea what the brain did? There you go. Either damaging it, although damaging is a little bit harsh, but what you have to remember is one of the greatest inventions in the history of the field of neuroscience is the invention of helmets. Because before helmets, young men went off to war and they died. After helmets, young men went off to war and they came back brain damaged. And the nice thing about someone who's brain damaged is they're missing a big chunk of their brain. And when they're missing a big chunk of their brain, you can see what it is that they're not able to do. And that tells you what the part of the brain they're missing they can do. I know this seems a little morbid, but it really is these types of observational things that helped us to understand how the brain works. One of the classic examples, and one that many of you have probably heard of, hold on a second. Sorry, it's heating up in my office now that it's getting into the afternoon and I'm starting to get the afternoon sun. So let me close up those windows. All right. One of the classic examples is an individual you may have heard of by the name of Phineas Gage. Phineas Gage was a foreman on the railroad. And one of the responsibilities of the foreman was to handle the dynamite. Right? When you were building the railroad, the goal was to get that railroad in as straight a line as possible. So when they came across, a mountain, you didn't want to build your railroad around the mountain, so you blew the mountain up. And the way they would do that is they would dig a hole, and then using these 10-foot tamping rods, these metal rods that were 10 feet long, they would put the dynamite in the hole and gently tap that rod to lower the dynamite to the correct depth so that when it blew up, it removed the rock and everybody was happy and they were able to continue to move forward. Well, one day, good old Phineas was tamping that rod down and the dynamite went off. And when the dynamite went off, that tamping rod came up, hit him in the forehead, and blew him back 10 feet into the air. Went through his skull, up out the top of his skull, and miraculously he survived. But what it did do was it destroyed his prefrontal cortex. And when it destroyed his prefrontal cortex, while he survived, his personality changed. If you look and read the reports, obviously as a foreman, he was a very mild-tempered, mild-mannered, respectable part of society. Yeah, and exactly. As someone mentioned, he basically became an a-hole. If you read some of the reports, he became the kind of man that would drink during the day and would curse at women, were some of the horrible things that he did because his behavior changed as a result of that, right? So things like him and what happened to him changed our understanding of how the brain worked and what the prefrontal cortex does. The prefrontal cortex is basically the location of our reasoning, our personality, and our intellect. I'm going to stop sharing this for a second because I'm going to try to find something. Again, we learned this from observing people like Phineas Gage. And then, as was mentioned, we would do research. Like, so instance, one of the things they would do is monkeys. Monkeys have a very hierarchical society in how they get along. And some are more aggressive, and if some of those more aggressive animals, what they would do is they would go in and inject acid into the prefrontal cortex. And as they injected that acid into the prefrontal cortex, they dissolved the prefrontal cortex, and shockingly, their behavior changed. All right? I know that's really shocking. Excellent. Here's what I wanted. Perfect. All right. Seeing yellow, so can I repeat that? Repeat what? I don't know what I was saying. I wasn't paying any attention. So, changing behavior. So it's personality. The prefrontal cortex is responsible for personality, intellect, and behavior. Now, what was remarkable about this was a remarkable finding at the time. Here is that half head chart we saw many times in the classroom. Remember, the nasal cavity is out here. And this ability, once we started to get an understanding of how the prefrontal cortex could influence personality and behavior, this was a remarkable finding at the time. Because up to that period of time, if you had a mental disorder, and of course there were many, right? You could have been a male who liked other males. Or maybe you were a female who just wasn't satisfied washing dishes and taking out the garbage and doing things along that line, right? If you were a little too uppity as a female, you could end up in an insane asylum, a sanitarium, right? For that poor behavior. And was the goal of putting these people who had these mental disorders into these places so that they could be healed? No, they basically put them in there so they wouldn't hurt themselves or anybody else. And they basically stayed there till they died. Well, now we started to get this understanding that if we could change the prefrontal cortex, we could change people's personalities. And so one of the things they eventually started to do is that they would take a large metal rod. And with that large metal rod, let me make myself a large metal rod here, they would insert into the eye, or what was commonly done was they would insert it into the nasal cavity. With that nasal cavity, they would then push that metal rod up through the cribriform plate into the prefrontal cortex. This was a hollow metal tube, and in the center of that hollow metal tube was razor wire, and there was a little slit in the end. And what they would do is they would then push that slit out so that a piece of razor wire would bulge out into that space, and then they would rotate that tube around, cutting all the connections to your prefrontal cortex. Of course, the patient had to be awake when this was being done, because it allowed you to see the change in their behavior. And that's how long they would do it for. They would continue to do this until, randomly cutting part the prefrontal cortex until their behavior changed. All right. Revolutionary finding at the time. There were some doctors that became quite rich and famous. One of the most famous ones was a doctor by the name of Watson. He had 2 of these canaliculi that he would put in his holsters, like 6 arms, like on his sides. And he would roam the countryside, going to different sanitariums, and he would compete against local doctors from speed. He could actually do both nostrils at the same time. And like I said, the people, not surprisingly, would come in nervous and anxious and aggravated, and they would keep doing this until they were calm. And once they were calm, they would stop. What did they call this process? Yeah, prefrontal lobotomy, absolutely. And it is absolutely as barbaric as I've described. Again, their hearts were in the right place, right? They wanted to be able to change people's behavior. And the ability to change someone's behavior so that they could be reintroduced into society is awesome. And it would change people's behavior. There are some people whose attitude and behavior was improved by this. But there are others where it was unaffected. There were others where it got worse. Infection was a big issue with many of these things, right? It wasn't consistent. It wasn't, accurate. It didn't do the same thing for everybody. It certainly wasn't accurate, right? And then there's, all the implications of breaking the nasal cavity into the cranial cavity and all these other issues. But as gruesome and as ineffectual as this is, why did lobotomies ultimately go away? Because we all got consciences and we all felt better and felt bad for these people and didn't do them anymore? No, the reason lobotomies went away is because we found something easier and cheaper. Why risk all this surgery and all this infection and have to have a specialist who comes across the country and I have to pay all this money for so that he can come and do that, when I can just give my patient this tiny little super cheap pill? I have no idea what this pill does. It helped Bob feel better. It doesn't seem to do anything for Susie. So maybe I'll give Susie two. Or maybe Susie, I'll give the blue pill and the green pill. I don't know what either of them to do, but when I put both of them together with Phil, Phil felt a little bit better. What we are doing today with medications is just as barbaric as what they were doing with the frontal lobotomies. Like the frontal lobotomy, we don't have any idea in many instances what it is that these medicines are actually doing. But God forbid little Timmy can't sit in his chair for 8 straight hours at school, so let's give him a pill so that he can behave the way we want him to behave. Hopefully 20 years from now, we will be talking about the medication of individuals the same way we talk about the lobotomizing of individuals, as a barbaric instance that is long past and that we don't talk about anymore. All right, I'll get off my soapbox now. But that gives you an idea of what the prefrontal cortex does. Yes, well, so yes, the mechanism by which many pills alter our behavior in many instances is not fully understood. Because it's not like we know that this particular nook of this particular gyrus happens to be where happiness is located. So if we could just stimulate the happiness, then we'll feel better. If we could lesion the sadness that's over here, we'll feel better. That's not how these things are stored. That's not how these things work. We don't fully understand how the prefrontal cortex works. We don't fully understand how these pills affect that behavior. We just know that they do affect the behavior. When I give Timmy the pill, he is able to sit down for 8 hours at school. And that's what his parents want, and that's what his teachers want. So that's what we do. And who cares what that pill is actually doing to Timmy, as long as it makes him behave the way we want him to behave. And exactly, and that's part of the problem. Again, I love that you guys are doing these privately for me, but it is a good thing if you let everybody see these questions. Yep, so again, one of the problems with it is, yes, different pills affect people differently. So it's not like one pill makes everybody happy. If you just give them one of them, everybody feels happy as a result of it. Different pills affect people different ways because we don't understand the mechanisms. So that's why I said, literally what we're doing with this medication is just as blind and just as barbaric. We don't know what the long-term implications are. We don't understand the short-term implications. All we know is the pill changes Billy's behavior, and that's all we care about. And that's part of the problem with medicine right now. All right. That is the prefrontal cortex. Like I said, I'll get off my soapbox now. Let's talk about a more fun area. That more fun area is this one right here. Notice it's Brodmann's areas 44 and 45. The way you can easily identify this area is, notice this particular area basically has 3 parallel gyri all next to each other. And this area, as you can see here, is what is known as Broca's area. It's called Broca's area because good old Bob Broca was the first person who identified it and described it. But the other thing it is called, and this might help you with that, is it's called the speech area. And if instead of Broca's area we call the speech area, you might be able to guess what the speech area does for you. What do you think the speech area does for you? There you go. It allows you to produce speech. Producing speech is a very specific and precise ability. Again, one of the ways we understand how these things work is when there's damage to it. People who have damage to Broca's area have what we call Broca's aphasia. It's a speech disorder where they're not able to produce meaningful speech. You can ask them to raise their hand. You can ask them to stand on one foot and hop. They can do all those things. But whereas I can sit here and say to you, Bill and I are going to go to the store to get milk, someone with Broca's aphasia would probably have to stutter through that like, Bill, store, milk. Not so much Tourette's, although that is a good question. Some Tourette's may be associated with Broca's area. Tourette's is a much more broad neurological disorder with many tics and things like that, so it isn't focused in Broca's the same way. But yeah, so it is exactly. So that's the second part of it, the stutter part of it. Absolutely. This is associated with very severe stutters. In fact, some people who have stutters, right? Anybody know someone who has a stutter or had stutter themselves? What's one of the tricks they teach people to try to get around that stutter? Anyone have a stutter or know someone who had a stutter? Know some of these tricks? There you go. That's the magic one I was looking for. Singing, absolutely. If you talk with a sing-song to your voice instead of a normal speaking pattern, you can often get around that, stutter that occurs when the person is trying to speak. You may not have thought of it in those ways, but singing really is a different ability than speech, right? What's her name? Who's the British chick that sings all the sad songs? Adele, right? Absolutely, excellent, right? Anyone ever heard her talk? She's got this big, huge, thick accent. Sounds like she's got a mouthful of marbles. You can barely understand what she's saying. But man, when she belts out those songs, her voice is crystal clear. She doesn't have that accent when she sings because accent is associated with speech. Singing is not associated with speech. So that's why sometimes using a sing song to the voice can kind of sometimes sneak around that, that speech deficit in that Broca's area, that aphasia in that Broca's area. One of the interesting things about Broca's area is that Broca's area typically I would say for right-handed people, over 90% of right-handed people have their Broca's area on the left side. About 75% of left-handed people have it on their right side. About 15% have it on the right, and about 10% have it bilateral on both. And they can actually test this a couple ways. Now they can do it by putting you in one of those fMRIs, functional magnetic resonance imaging, and they can look at blood flow to parts of your brain while you do activities. So what happens is they have you speak, and they look at which side of your brain lights up. But back in ancient times, and by ancient times, I mean when I was in graduate school, what they would actually do is they would inject an anesthetic into one side of your brain. Your carotid arteries, for the most part, your left carotid artery feeds the left side of your brain, and the right carotid arteries feeds the right side of your brain. So they would inject an anesthetic into your left carotid artery, anesthetize the left side of your brain, and then ask you to say your name. And if you couldn't say your name, it's because they had anesthetized your Broca's area. For those people that have it lateralized, typically on the other side is more mathematical abilities. So mathematical composition. So on one side it's speech, on the other side it's mathematical composition. All right. One more frontal lobe region. And that last frontal lobe region, and this is where it gets a little tricky, so I want to make sure to emphasize this. Notice the structure I am highlighting right now. The structure that I'm highlighting is, of course, the Precentral Gyrus. That is the structure that I just highlighted around. That is the structure. However, on that particular structure, the cells on there have a specific function. So that area of cortex, that area or region of cortex, has a specific name as well, and it is the primary motor cortex. So in this case, you have a structure, and that structure has a function, that area or a region on it. And that area or region on it is the primary motor cortex. And guess what the primary motor cortex does? Come on, primary motor cortex. Movement and more movement, motor, I love those. Those are correct answers. But remember, at this point of the class, we want to start being more specific. What our primary motor cortex does is it controls the voluntary movements of our contralateral skeletal muscle, right? Voluntary control over skeletal muscle. Remember, contralateral is a word we learned way back at the beginning of the class, which means opposite side. So notice here, we are looking at the left primary motor cortex, it would voluntarily control all the skeletal muscle on the right side of the body, which you knew. You knew the right side of your brain controlled the left side of your body, and the left side of your brain controlled the right side of your body. Here we've just said it with fancy words. So sane motor and movement is correct, but if we talked about identifying the function of that, and that's all you put, motor or movement, you're not going to get full credit. For full credit, we need voluntary control of the contralateral skeletal muscle. All right. Questions on that? All right, that is how the game is played. Let's do one more before we take our next break. So any questions on the 3 areas of our frontal lobe? All right, well then let's talk about and actually I'm gonna leave that there because that's useful for me. Let's talk about the structures, the regions, I should say, of our of our parietal lobe, and how many of those are there? How many parietal lobe areas or regions do we need to know? Just 2, that's not too bad, excellent. So we are now talking about our parietal lobe. So again, that's basically from here to about here. This is our parietal lobe. And I'm gonna change the color of my highlighter now. Let's use, I guess I have to use orange maybe. Does that show up? There you go. Our first area is again on a structure. So let's do it the same way. Notice the structure that we highlighted is our post-central gyrus. And the gray matter, the cell bodies in that area or region of cortex is called the primary somatosensory cortex. And guess what the primary somatosensory cortex does? Somato, you know what somato refers to? Soma refers to touch. Absolutely, that's exactly what it has to do to. This is where we process the information of touch from the contralateral side of the body. So if someone were to rub the back of your right hand, then you would feel it here on your primary somatosensory cortex. If someone was to pull a hair out of your leg, because again, like me, right, with us all at home, I'm not shaving my legs right now. So it'd be easy to grab a hair and pull it out, and I would feel that, right? That contralateral sensory. Yes. Was there a question? Okay, guess not. Excellent. Now, remember the other thing we talked about is there is some specialization to this. I think for this, I think I can write in white. What's cool about both the motor and the sensory areas is there is specialization. Down here on both is where we have the motor control and the sensory sensation of the face. Then up here is where we have the hand. And then up here is where we have the trunk. And up here is where we have the legs. Okay. We have this very precise map of our body on this. And you may not have thought of it in these terms, but you're probably aware of it because there's a big, large blood vessel that goes down the central sulcus. And if that blood vessel gets blocked, one of the things that can happen is that blood can stop flowing to a specific region of this cortex. We call that condition a stroke. And when that occurs, that tissue gets damaged. And when that tissue gets damaged, it doesn't function anymore. So maybe grandpa had a stroke and now he's lost both the movement of his hand and the sensation of his hand. But the maps aren't truly identical, so maybe he can't feel any, or he can't move anything up to the shoulder. His whole entire arm has been paralyzed, but maybe he can feel from the elbow up. Maybe he's just lost feeling from the forearm and the hand. So the two maps aren't identical, and the damage won't be identical as well. So maybe some of the arm has been salvaged here while all the arm is lost over here. So often those types of strokes affect both movement and tactile sensation because those two maps are right across the bridge from each other there. All right. Questions on our primary somatosensory cortex. All right, the last area that I want to identify, and I'm going to highlight it here first, but then I've got a couple more pictures I want to show you. Highlighter, go stick with orange. Notice here, they've drawn it as this dotted line here. And notice it is an area known as Wernicke's area, because good old Bob Wernicke was the first one who described it. But notice if we look at a couple other pictures, clear the drawings from that one, this one here. Notice here, Wernicke's is, oops, some of 39, 40, even down here to 22, a little bit, something like that. And we clear that, and notice the next picture here. Notice it even covers a little bit of lateral sulcus. So this one is a little bit more of a general area. It's called Wernicke's area, but notice it has another name as well, and that other name as well is the general interpretive area. Wernicke's area, or this general interpretive area, is where we make sense of speech. Oops, and I can't use white for that anymore. Notice not where we process speech. Remember, process speech is over here in the Broca's area, right? This is Broca's area where we process. This is where we make speech. This is where we make sense of speech, right? This person who has a problem here stutters or can't speak at all. This person has no problem speaking, but they have difficulty speaking, not even articulation, but they have problems in speaking words that make sense. Remember, this person can't speak, but they can understand language. You tell them to raise their arm, and they raise their arm. This person may not be able to do this. And again, as I said, one of the best ways to be able to distinguish these is to distinguish these in people who have damage to this area. People who have damage to this area are called Wernicke's, have what we call Wernicke's aphasia. And if you do a YouTube search, you can find many examples of this, but I want to show you my favorite. Where is it? It's here. Come on. There we go.
Their foot, you know, boom.
What's this?
You know, the big anchor right there.
Can you guys hear that?
Really
I'm sorry?
clear, like maybe captions.
I don't know if there's captions for it. Let me try one more thing. Share computer sound. See if this sounds better.
All right, end of the football.
End of the football?
It influenced the football right here.
Do you guys hear that? Influence the football right here, end of the football? You guys can hear that okay? Okay, let's listen and let's watch and listen.
Cause it comes in, see?
Yeah.
The end of it, see? Yeah. Oh, we have a lot of things, see?
I don't understand.
Well, it's pliable in the other, see? You don't understand it, but it applies in the long reed comes from there. And it comes source from the country out through it, see? All the under it, see, it comes up and finally come out here and goes out, see? Get it?
Sort of.
Yeah, it comes out here, see? It comes out here, and here. And it comes in, and it comes out, and bing. That's a good flow, see?
That's a good what?
That's a good flow. Flow goes to fire, see? It flows cold or it gives or kicks out or bing or whatever it is. We has no football far out. It's just pulling, see? But they say feel it. Oh, I get you when you have bed, see? I don't know.
Where do you live?
Where do you live? You live? You're up here, see?
Where do you live, Doc?
Where do you live?
Where do you live?
Where do you live is, well, if there's a run here, around here. You?
You used to be a dentist.
Yes.
Yeah. Where?
Right here. the rest of the here, yes.
Yeah. How many years were you a dentist?
How many? Holy Christmas. Oh, about 100 and, let's see, 130, about 40, no. Gosh, what time is it? 1 plus 5, wait a minute, no. It's a pretty hard class to get when you get to this one day. It's about every year off, see. It's claws, it goes for about over 30, see, or 130 for down, see, in front of out for it goes straight against her, and then it comes back to the against her to 4, and then goes out for the rest of the time. The third is not the servant, but the soldiers, where they have the soldiers of the Sandbirds and the Cerebrums and all the first Cerebellum, and watch out in the short side, and they cross with the sugar 2 hours, 2 low. Wowie. God, these are terrible. Wow, these are terrible words.
These are terrible words.
They sure are.
Hard?
Yes.
What you're saying, you mean?
Yeah, that's what they do. They actually do them a lot harder. It's when they outer in hard go. And then when they go out, finally they quit, see?
So notice if you were sitting at a Starbucks and these were the two people behind you, you wouldn't necessarily think twice about what you were hearing. It sounds like they are having a normal conversation. It isn't, it's just not, hold on, stop sharing that, come back to this. It isn't till you actually start listening to what he's saying that you can see that it isn't making a lot of sense. Now, again, some of you guys had some really good comments on this. It does sound like maybe he's a little drunk, you know, or having struggling with his words that way. And the $64,000 question is as to whether or not they can understand what's being said to them. Obviously, clearly, there's some parts of what they're talking about that he understands. And I've watched this video many, many times. So she asks him, right, you know, you were a dentist. And then she asks where, and he's all here and points at his mouth. So clearly he understood that and even made a joke. But then she asks him how long he's been a dentist for, right? And he's trying to explain that. And I've watched this video enough to that I think I understand what he's trying to say. What he's trying to say there, if you're watching in that whole thing, is that he went to school, he became a dentist for a few years, and then had to go away to war. Right, he's talking about the soldiers and around the backside and all of those things. And he was at war for a period of time. And then he came back and was a dentist for the rest of that time. And I've gleaned this information from watching it many times. So clearly he understands what she asked and he's trying to answer it. But remember, there were other times he asked her, where do you live? Right, if we were sitting in the classroom and I looked at you and I said, where do you live? What you likely would say out loud is, where do I live? Because you're processing that information. Ooh, do I really want him to know where I live? Should I give him general area? Should I give him state? Should I give him city? Should I give him my home address? You're thinking and processing it. And notice when you're thinking and processing it, you turn that question around. I ask you, where do you live? And you say, where do I live? But when she asks him, where do you live? He repeats the question exactly. She says, where do you live? And he says, where do you live? He's saying the words again because he's trying to make sense of them. He thinks maybe she's saying lips because he eventually points at his lips. And then my favorite part of this video is at the end. He clearly understands some things, and exactly, that's the key at the end, right? It seems like he's saying, I can formulate the words, I can say the words on the inside, but when it comes to them coming out, they just die, they quit, right? And so clearly he understands that he's having some deficiency. But how much he understands what he's saying doesn't make sense, or what does make sense, and how much he understands. Part of the problem with this disorder is that lack of ability to communicate. So it is hard to be able to fully understand what they understand and what they don't understand about their condition. And that's also a great question. Some of these people, depending on the damage, some people it doesn't affect writing, but in other people it does. And here's the coolest part of all of this. This general interpretive, again, is a localized ability. So I lost my annotation tone. So again, for almost everybody, this general interpretive is, this Wernicke's area is on the left side only. And what's really cool is on the right side, it is spatial awareness. Oh, where am I writing that? I don't know where that went. There we go. Oh, no. On the right side, it is spatial awareness. So what happens is if people have injuries over here on the left side, they get that Wernicke's aphasia, or what some people commonly refer to as word salad, that Wernicke's aphasia. If instead you have the damage on the right side, then what happens is you start to ignore the left side of the world. So damage here, you start to ignore the contralateral side of the world. So you show them a potted plant and you ask them to draw the potted plant, and here's what they draw. They just draw the right side of it. When they get dressed in the morning, they will only put clothes on the right side of their body. They'll forget to dress the left side of their body. They'll forget to wash the left side of their body when they're in the shower. They will get pissed at that dude that is in their space by getting so close to them, and get angry at the left side of their body for being too close and being in their personal space. They ignore the left side of the world because of that change and understanding of spatial awareness. Again, this stuff, it's like I said, often, and this, one of the things I love about the field of neuroscience is the diversity of it and the interest of it. And it's very hard for me not to get into the academia. Obviously, clearly, if someone has this deficiency, I had a student, 2 years ago now, whose boyfriend was in a car accident and had damage to the right side of his general interpretive area, and so lost that spatial awareness. And so he was one of those people who would forget to put the shirt on the left side of his body, or would forget to wash the left side of his body. And again, I appreciate for those people that it is very challenging, but from an academic standpoint, these things are really cool, how different and amazing and diverse the brain and its functions are. All right, questions on that? All right, we are through 2 of our lobes. Let's go ahead and take our second break here. We will go ahead and take another 10-minute break. So let us return, what's my clock say, 2:41. So let's come back at 2:51. And at 2:51, I will start the recording. And we'll come back then. All right. Any questions before I walk away? I saw last time I walked away and then someone asked a question. So I'll give you guys an extra second.
Yeah, I was curious. Is there any chance that you can stay on a little after?
To be able to answer questions? Sure. If you have specific questions, I can do office hours after. Absolutely. Happy to do that.
Thank you. My kids kind of distracted me and I got lost.
Okay, well, no problem.
All
All
right.
right, so let us go ahead and. So, great question here. So real quickly before we go, there's a couple good questions here. You are correct that some of these pictures, the Wernicke's area is slightly different. So what they represent as Wernicke's area, what's represented as Wernicke's area here, what's represented by Wernicke's area here are all slightly different. But notice all of them, basically it's at the end of the lateral sulcus. So if you follow the lateral sulcus to its end, that's where Wernicke's area. Follow the lateral sulcus to its end, right? Even here, follow the lateral sulcus to its end, and that's Wernicke's area. So that's really going to be the key to finding Wernicke's area. I will put the tag here. I can use any of these pictures to represent it. So I could do any of those to be able to represent it. So again, don't try to just memorize the pictures, but know that that is how we will try to interpret it. So yeah, and as I mentioned, I'm always happy to answer questions after the official lecture time is open for anybody who wants to say. I usually come a few minutes early, but also if someone wants to meet earlier, I can do. We don't have official office hours anymore because I'm pretty much on call most of the time, but I'm happy to stick around before or come early if anybody has or needs extra help or has questions. And then I can always meet outside of normal class times as well. All right. We are almost halfway through our lobes. So the next lobe, we finished the parietal, we finished the frontal. The next lobe we are going to look at is the occipital lobe. And the occipital lobe is where the visual system is located. Again, I've said at the beginning of the class, and I'm not shy about it, I am a visual neuroanatomist by trade. That is what I got my PhD in, is studying the development of the visual system. So I am entirely biased. I love the visual system and everything about it. It is the best thing in the whole wide world. However, my bias aside, we are still visual organisms, and your visual system is your most important organ system. I know it's not an organ system, I don't care. It's the most important thing. Your retina, has trillions of cells to perceive light, as we talked about. The optic nerve that carries the information from your eyeball to your brain, each optic nerve has over 1 million axons in it. Over 40% of the information you take in from the world around you is visual information. Over 60% of your cerebral cortex is involved with processing visual information. We are visual organisms. And here in the occipital lobe is where that processing begins. As I mentioned, it starts right here. And let's switch to my highlighter again. The very back of the brain, what Brodmann called area 17, is our primary visual cortex. And as the name indicates, it is the first region where we perceive light. Again, if we think about deficiencies and how they would work, if you have damage to your primary visual cortex, you no longer see light. Everything is dark. Everything is black. Hopefully, nobody here has a lesion to their primary visual area. However, you might have taken a blow or two to the head before. Area, cortex, region, all of those are interchangeable words. When I say area, I mean area of the cortex. When I say region, I say region of the cortex. So yes, we're talking about a portion of the cortex, area, region of the cortex, all means the same thing, yes. Maybe you've taken a blow to the head. Maybe you've taken a fall. And as there was that jerking sensation of your head, while there are all these protections for your brain, one of the things that may have happened is the back of your brain may have been pushed against the back of your skull. And when that happens, what occurs to you? You see stars. There you go. That seeing of the stars is that irritation to the primary visual cortex, where you see those stars. You see that light because you have stimulated in a unique way, but you've still stimulated those nerves, called them to fire an action potential, and you perceive light. So that is where you perceive light. The rest of this, and let's change colors, is what we call visual association area. The primary visual cortex is where we get the raw data. In this case, that raw data is just light. So damage there, do you see light? Do you not see light? The association cortex is where we process the information, where we make sense of it, of that information. And some of it can be more general. Some of it can be really specific. And again, one of these ways that we understand that is by deficiencies people have. For instance, area 18. And again, here's what I'll say. For the exam, you just need to know that visual association cortex makes sense of the information, processes the information. But I am going to give you some examples of that just so you can, again, understand how this works. Usually when your eyes get blurry from standing up too fast, it's more of an almost lack of drop of consciousness because of the change in blood flow to your brain. Indigo? I'm not sure what you mean by indigo. Oh, is that what they call seeing stars? Oh, vertigo. Oh, okay. Yeah. Oh, vertigo. Oh, got it. Okay. Yeah. Yeah. No, vertigo is either a balance thing from your inner ear, or it could be a drop in blood pressure thing. All right. So area 18 is where you process color. Now, I'm not talking about like red and green color blindness. That's something in the retina of the eye. What I'm talking about here is if your area 18 is damaged, then you're in a black and white film. All right. And what's really cool is, like I said, over 60% of our cortex is involved in processing visual information. We have these two big pathways where there's large amounts of this association cortex where we make sense of information. In fact, what we have here are two pathways. This up here going towards the parietal lobe is what we call the where pathway. And this one going towards the temporal lobe is the what pathway. And I will give you two examples of how this works. In the where pathway, you have an area, and I'm not even going to write this area down because you don't need to know the name of it, but there is an area known as MST, 3 letters, MST. MST is the part of your brain that perceives optic flow. What that mean by that is if you sit in your chair, and if you're like me in an office chair, you can spin it around. And as you spin it around, you see the world moving from left to right or right to left, depending on which way you spin. But also, if you stand up and walk in a straight line, as you stand up and walk in the straight line, if you think about what's happening, the world starts at a center point in front of you and then kind of comes and flows past you. What happens with people who have damage to MST is they don't perceive that optic flow. Now, again, I doubt anybody in the class has this deficiency, but how many people have ever been in a room with a strobe light before? Anyone ever been in a room with a strobe light before? Absolutely. If you think about it, if you watch someone wave their hand, you don't see the continuous movement of your hand. First, their hands to the left, then their hands in the middle, then instantly their hands to the right, and then back in the middle and back to the left. You don't see flow, you see a bunch of static images that constantly change. That's what it's like if you have damage to your MST. If you have damage to your MST, not the traces so much, but what I mean is the static images, first, second, third, instead of a continuous movement of the hand. That person can't drive. That person can't pour a cup of coffee for themselves because first the coffee is quarter full, then it's a half full, and then suddenly it's overflowing. However, it turns out these people do pour coffee for themselves because they put their thumb in the cup, right? If you think about it, if you put your thumb in your cup, you could pour the cup of coffee blindfolded, right? Just pour until it touches your thumb and then you're fine. So again, we have ways of getting around there, but they live their lives like they're constantly at that rave and constantly in that strobe light. In the what pathway, that's the where pathway, where something is and how it moves through space. The what pathway involves identifying what an object is. There's an area called IT. IT in the what pathway. And what again, it stands for interotemporal. So I know seems like it tells you what it is. But what its job is to tell you what something is. It is. There's a famous case of an individual who has damage to his IT area. And in one of the classic cases, what they did is they showed him a glove. It was a knit glove, and he was able to describe it perfectly. It was blue and white striped. It had the processes that came off of a central region. He could describe it perfectly. But then they asked him what it was. And he thought about it and thought about it and thought about it. And finally he said, he thought it was a change purse. Because as he looked at the fingers of it, he thought, oh, one of those must be for quarters, one of those for nickels, one of those for dimes, one of those for pennies. So even though he could see it clearly, he didn't know what the object was until they put it in his hand. When they put it in his hand and he felt it, then he was able to realize that it was a glove. And that's a great next question. No, he cannot recognize faces either. He could be sitting there in a room and his wife could be sitting right next to him and he will not recognize his wife. If she gets up and moves, sometimes her movement, the way she walks, he can recognize her that way. And of course, the second she speaks, he can recognize her as well. But if she just stands still, he does not recognize her. There have been actually many books written about this individual. One of them was The Man Who Mistook His Wife for a Hat. It's the story about this guy who has this IT deficiency and is not able to identify what items are. So again, some of these brain regions are incredibly complicated in their processing. But all of these things, as fun as they all fall into that visual association cortex, right? So we have the raw data that comes in the primary. We have the making sense of that raw data. That is what the function of the association cortex is. And we have that for the visual system in the occipital lobe. And if you've peeked ahead, you can see we have the exact same thing in the temporal lobe for hearing. For hearing, here, right on the lateral sulcus. And notice basically the central sulcus almost points right at it. This picture doesn't show it as well. There's some other pictures. Let's take a different picture. There. So again, notice it points almost right at it. This area, the central sulcus almost points right at it. I like that other picture better. Let's go with that one since I drew it. Central sulcus kind of points right to the edge of it. The darker region right on this, that is the primary auditory area. And this is where we get the raw sound info. Yes, vision expands beyond the occipital lobe. Remember, as I said, over 60% of your cerebral cortex is involved in processing visual information. Primary auditory area is on the temporal lobe, right on the lateral sulcus. It's where we get the raw sound information. And then again, the association area, this area out here, is where we process that information. We make sense of it, process sound. So the primary auditory is where we get the sound. The association cortex is where we see that it is an oscillating sound that is increasing in intensity of in amplitude as it's getting closer to us. And so we realize it is a siren of an ambulance moving towards us. We should step out of the way. Yes, if he touches his wife, then often he can recognize her that way as well. So touch, sound, and like I said, even sometimes movement, he is able to recognize her. But just viewing her, just looking at her, he cannot recognize her. Yeah. And like I said, that's what happened with the glove. Once they put the glove in his hand, you could feel it. He instantly realized, oh, this is a glove. He was able to make that connection. But when he was just looking at it visually, he couldn't connect what he was seeing with his understanding of the objects of the world around him. And like I said, auditory works the same way, only on the temporal lobe. So those are our 4 lobes of the cerebrum. However, as I not so subtly mumbled when we went through it the first time, they're really 4.5 lobes. And that's because if you take the lateral sulcus and open it up, and as we saw it before, hold on a second, let me answer. Okay, I'll answer that question first. For the auditory cortex, Most people who are deaf or have hearing losses, it is more damage to the ear or to the inner ear, you know, the sensory structures that perceive it. It is not typically cortical damage where they have that hearing loss. So most deafnesses, although there are some deafnesses related to cortical damage, some strokes that can cause it and things along those lines. But most types of deafness have more to do with the sensory structures that perceive sound as opposed to our processing of the sound. And again, if you think about it, that's what they do with a cortical implant. Many people who are born deaf, the problems are with their cortex or with their ear, the perception of it. So what do? They basically put a microphone behind the ear and they connect that microphone directly to the cortex. So now you have a different signal that is receiving the sensory information, receiving the sound, carrying that sound to the brain. And once it carries sound to the brain is able to process that information and make sense of it. That's basically how those cortical implants work. They bypass the inner ear, the sensory structures, and instead they carry that information straight to the brain. If the deafness was caused by damage to the brain, then that cortical implant wouldn't help because the problem was the brain problem. And so there's nothing you could do really at that point. Great questions. I'm sorry for plowing ahead. Any other questions before we continue? Just, I see we continue to fall more and more behind, so that's why I'm trying to push through a little bit more. All right, excellent. So, as I mentioned, there are really what are sometimes thought of as a fifth lobe located inside of that lateral sulcus. Here's the frontal lobe, here's the temporal lobe, here's the lateral sulcus. When you open up the lateral sulcus, there's some extra gray matter in it. We see this great picture from your textbook that shows it, but the other way we show it really nicely, and if you notice, we saw it at the very beginning, was in this coronal section. Notice here in the coronal section, this is the parietal lobe up here. This is the temporal lobe down here. This right here is the lateral sulcus. And notice with the lateral sulcus, what color am I going to use? Green. In the lateral sulcus, this part in here is gray matter. It's an extension of the cortex, but it comes deep inside of the lateral sulcus. And so this region is sometimes referred to as a fifth lobe. So there are some individuals that refer to this as a fifth lobe, but what this structure is, whether it's a fifth lobe or not, the cortex inside the lateral sulcus is called the insula. The insula is a very important piece of cortex. As we're learning more and more about the insula, we know that the insula plays an important role in our processing of anger, pain, and two of the other important ones are desire. Right? This desire is related to, we're finding more and more, this is related to addiction. One of the really interesting studies that they have done is they will take, for instance, gambling addicts and people who are not gambling addicts and put them in one of those fMRIs we talked about where they can look at blood flow to different parts of the brain. And what they'll do is they'll have them play games of chance. Well, they'll have to guess whether a card is going to be black or red, right? Or guess the suit of the card or something like that. But usually it's something as simple as black or red. Is the card going to be black? So is it a spade or a club, or is it red, a heart or a diamond? And what they find is that in those gambling addicts, they get a much higher activation of the cortex here in the insula. This insula lights up when they're doing these games of chance, whereas someone who isn't a gambling addict doesn't show that same activation in the insula. They've done similar things with sex addicts. They show erotic pictures to them, and some people light up in this area and others don't. right? Alcohol, although those are trickier to do, but what they're finding is this area, the insula, may play an important role in our desire, and that strength of the desire for those activities may play a role in addiction, right? So in an ideal world, if you think about it, if you were to go in and suppress these neurons so that they didn't fire when they did the activity, they wouldn't get that same desire, and it could biologically be possible to break someone's addiction just by being able to dampen the firing of their neurons, right? Again, we are not anywhere near being close to being able to do something like that, but this type of research is leading to that type of potential. The other important thing that our insula does is, remember we talked about all these nuclei, these clusters of cell bodies that are down here. These clusters of cell bodies collectively have a name. This collection of nuclei inside, or let's say it this way, deep in the cerebrum, is called the limbic system. And our insula is the cortex that controls maybe isn't the right word, but let's say it this way. It's the area of the cortex. The insula is the area of the cortex that is associated with the limbic system. So like I said, it plays a role in anger, pain, desire, and addiction, but it also is associated with this limbic system. And I want to write this down, my definition that I've got here of the limbic system, because I like it and I want to make sure we understand it. The limbic system, as I said, is a collection of nuclei. That's these nuclei that we see here. And it is a functional group. Right? They work together towards the same goals. The types of things that they work on is memory, storage, and retrieval. Emotions. And here's the part that I wrote down because I wanted to make sure I got this precisely. It links our conscious thought to our unconscious functions. Ooh, that sounds fancy, right? We can call Grandma on the phone and say, Grandma, guess what? Our limbic system links our conscious thought to our unconscious functions. And she'll be very impressed, and she'll send you 20 bucks in the mail. But what does it mean? Well, you know what it means. It's just a fancy way of saying this. When you come stumbling home drunk, back in ancient times when you used to be able to go out to bars and get drunk, when you came stumbling home drunk and was fumbling with the door trying to get the lock unlocked, making a tremendous amount of noise and waking the dog and waking the baby, and your loved one comes to the door to open up the door and let you in because you keep fumbling with the keys, as you look at that loved one and you see that blood vessel on their forehead pulsating, right? You can just tell how excited they are to see you and practically aroused and ready to take you to bed, right? No, our emotions, and that's our emotions, what we're thinking affects our body physiologically. When you are angry, when you are sad, right? When you're hungry, when you're aroused, when you're scared, all of those emotions are expressed physiologically, right? What you think about affects how your body behaves. If you're in pain, your heart rate goes up, your temperature goes up. Right? This is the kind of thing they try to take advantage of with not muscle memory. We'll talk about that in a minute, although there are other parts of the brain that do that. No, what I'm talking about here is like what they use in a lie detector test. In a lie detector test, one of the things that they do is they sit you down in that chair and they ask you a bunch of benign questions. What is your middle name? Who was your first pet? What is your favorite color? Where were you on the night of the 14th? Right? Their goal is to make you think about that bad thing that you did. And when you're thinking about that bad thing that you do, you sweat a little bit more. Your heart rate goes up a little bit. Your blood pressure goes up. The things you think about affect your body physiologically. And it's the limbic system that does that. This limbic system links the things we think about, our memories, our emotions, and those types of things with our physiology of our body. Changes our heart rate, changes our blood pressure, dilates our eyes, dilates our blood vessels, all those types of things. That's what our limbic system does. Now, if you notice, here we see some of these things in a cross-section view. Here is another great view of this as well. So this is, I'm sorry, not cross, this is a coronal section. Here we have a partial sagittal section, and then we see some of these things intact, so we can see the structures, the white matter structures, the axons that connect these things together. So notice, for instance, as we look at this, here we can see some of the structures, starting first with this purple structure right here. This purple structure off here to the side, and this is what we see down here, kind of in the thumb of our temporal lobe, is a structure that is known as the amygdala, or the amygdaloid body. The amygdala is a nucleus that relates memory and emotional reaction. In particular, the amygdala affects anger and fear. In our house, this is what we refer to as the worry monster. My daughter, when she was young, had a very active amygdala. Your amygdala is your worry monster. Being able to recognize fearful situations or to get angry is important from an evolutionary standpoint, right? Back in ancient times, we used to live in trees, right? And bears with axes roamed the countryside. So when you heard a noise, it was likely a bear with an axe, and you needed to be scared. However, if it's 2010 and you're 3 years old and you're nestled into your bed in your gated community and you hear a little creaking noise out in the hallway, it is very unlikely that it is a bear with an axe. But that's not what your amygdala thinks. Your amygdala still tells you that that's a bear with an axe, or there's a monster under the bed, or there's a monster in the closet. Your amygdala's job is to take an unlikely fear and elevate it. Right? That's what it does. It gives you that fear response. And it's important for survival, evolutionarily. Maybe not so important nowadays, but it still, unfortunately, can be quite active, right? And the other way. Again, one of the ways that we see this is people who have deficiencies in them. One of the great examples are in monkeys. A lot of this research is done in monkeys. Monkeys are incredibly social individuals. And one of the main ways monkeys communicate is with facial expressions. So one of the things a monkey will do is they will sit in a chair and you can show them pictures on a computer screen, kind of like what you're doing right now. And they basically will show them angry faces and submissive faces. And they're very good at recognizing which is which. So they've got a button to push, and if they see an angry face, they're supposed to push the button. And if they see a submissive face, they don't push the button. And what happens is every time they get a picture, they get rewarded with a little bit of juice. So they get a little sip of juice, and they will happily sit there and do this activity for hours. And they're really, really good at it. Unless their amygdala is damaged. If their amygdala is damaged, they can no longer tell a difference between the submissive face and the angry faces. Right? So that's our worry monster. That's that amygdala. There's a relationship of memory and that emotional reaction, especially when it deals with fear and with anger. Another important structure, we don't see it so much in this picture, but let's switch to this picture here. Notice this picture here, we have this lateral structure that is a red structure out here in the thumb of the, again, lateral in the thumb of our temporal lobe. And this is called the hippocampus. The reason it's called the hippocampus, this picture doesn't do it service. The reason it's called the hippocampus is the hippocampus here and I'll just draw it out here in symmetry, has this elongated body, and then this round part here, it actually curls up. It's got a curled part in there. And that's actually where it gets its name. Hippocampus means seahorse. So basically this thing has a body, has a tail like a seahorse, so it looked like a seahorse, so they call it the hippocampus. This hippocampus is where we convert short-term memory into long-term memory. All right. I can give you 3 objects: cat, ball, truck. Someone repeat them back to me.
Cat, ball, truck.
Excellent, right? Hopefully the rest of you are able to remember that as well. You were able to do that, and someone who has damage to their hippocampus is able to do that as well. However, then I continue to talk for a couple minutes, or better yet, I give you a bunch of mathematical questions. What's 2 plus 2? What's 5 plus 3? What's 7 minus 9? What's 8 plus 5? What's 14 times 362? I give you all these basic mathematical questions. So you do that, and then I ask you again, what were the 3 objects that I had asked you to identify?
I only remember ball and truck.
Anyone remember them all? There you go. Cat, ball, and truck. Absolutely, right? If someone who has damage to their hippocampus wouldn't remember those 3 objects, not even 2 of them. They have working memory. Working memory is when you're holding it in the front of your brain, thinking about it consciously. But as soon as you get distracted by something else, like mathematical equations, if you're not able to put that into long-term memory, you can't recall that information. And so that's what your hippocampus does. Your hippocampus converts short-term memory into long-term memory. There is a very, very famous individual by the name of, he's only known as subject HM. He was a young man, early 20s, camping in the woods. And it started to rain. So it was either a kerosene barbecue or charcoal or something like that. It started raining, so he decided to sleep in his truck. And he slept in his truck with some type of barbecuing equipment. And as a result of that, he got carbon monoxide poisoning. He survived, but he damaged, his brain was deprived of oxygen for a brief period of time because of the carbon monoxide poisoning, and he has bilateral damage to his hippocampus on both sides. And since that event, which now occurred probably 40 some years ago, ever since that event, he can no longer make long-term memories. All right? There is, at UC Davis, at the time when I was at UC Davis, one of the instructors there was one of the first researchers who worked with him. And at the time, while I was there working at UC Davis, they hit the 25th anniversary of his deficiency and of this instructor working with him, and he wrote a paper on it. And again, at that time, the guy had been working with him for 25 years. There you go. That's a perfect example. We've been working with him for 25 years at that point. And every single morning when he went in to talk to him, he had to introduce himself for the first time. He'd leave to go to lunch and come back, and he'd have to introduce himself to him again. All right, the guy's been working with him for 25 years. He has no memory of him. There are actually 2 excellent movies involving problems with hippocampus damage. 50 First Dates is the comedy that is with that. Excellent movie, great depiction, for the most part, accurate depiction. All the people in the Memory Institute, the goldfish guy and all those aren't realistic, but they did a pretty good job with her. But anyone know what the other movie? The other movie was a classic thriller movie. Anyone know the second movie? Excellent movie involving someone who had short-term to long-term memory deficiencies. It's a very long time ago. Guy Pearce, Memento. A story of a policeman who a criminal came in and attacked him and his wife, killed his wife. He lost a lot of blood, ended up brain damaged as a result of it, and he wants to get revenge. But he cannot convert short-term memory into long-term memory. So he has to write notes, take pictures. He gets tattoos on himself of important pieces of information as he tries to track down the person who killed his wife in this situation. And so excellent movie. If you haven't seen it, I highly recommend it. Excellent, excellent movie. But that's what it's like. And people like HM, we have learned a lot about memory. Obviously, this guy's got a whole lot of time on his hands. So one of the things, for instance, he does is play a lot of video games. And this is where the thing about muscle memory came in. You can hand him a cartridge for a video game and ask him, have you ever played this game before? No, I've never played this game before. He has no memory of playing the game before. But as soon as he puts the game in and starts playing, he'll get the high score. Because even though he doesn't consciously remember playing it, that muscle memory of moving the fingers and moving the joints to be able to move your man to do the shooting or do whatever, that memory is retained. And so he actually has that muscle memory from the activity, and that's outside of the cognitive working memory. Again, 25 years have gone by. He obviously, when he wakes up and looks in the mirror, it depends. So as long as he keeps the information in his working memory, if he were to sit there and think, right, cat, ball, truck, cat, ball, truck, cat, ball, truck, he'd be able to retain that information for a period of time. But as soon as he got distracted, oh, look at that bird that went by. What would you like for lunch today, ham or cheese? Oh, I don't know. Do I want a grilled cheese? Do I want a ham sandwich? Boom, that information is gone. As soon as it's out of his working memory, it's gone and he's lost it. He obviously, 10, 15 years even into this process, when you wake up first thing in the morning and you have no new memories, the last thing you remember was camping. When you look in the mirror, you don't see the person you expect to see, right? And so you would expect maybe some more surprise or shock. Now, again, if you look at the image down here, you can see that the hippocampus is very close to the amygdala, and his amygdala was also damaged. So he is also very muted in his emotions as well. So he clearly knows as he's shaving that that's him that he's looking at in the mirror. He has a sense that time has gone by and that is something has happened to him, but he doesn't have any real sense of how much time has gone by or really the state or understanding of what his condition is. All right. Questions on that one? All right. Notice now there are some other important structures. What's next on the list? Okay, excellent. This picture here. Notice on this picture here, and we'll talk about this next class. This structure here is the olfactory bulb, and coming off of the olfactory bulb are a bunch of axons that form our first cranial nerve. This is our olfactory nerve. And of course, our olfactory nerve is what gives us our sense of smell. One of the interesting things about our sense of smell, and there is two of them, but one of the interesting things about our sense of smell is notice this olfactory bulb, this olfactory tract, brings sense of smell information straight to our limbic system. You may not have thought of it in these terms, but you are aware of this because our sense of smell is the one that is most closely related to memory. If you're like me and you think back to that one guy who got away, when you're feeling really sad, you look at the picture of it. When you're feeling a little sadder, you listen to that last recording you have of him on your phone, that voice message that he left. But when you're really feeling depressed and miss him, you grab that shirt of his that you have in that Ziploc bag and you open it up and take a nice big whiff of it. And that smell of his aftershave or his cologne or his body odor brings all those emotions back. Or maybe the smell of freshly baking bread reminds you of that summer you spent at grandma's house, and you can almost feel the grass under your feet, or the feel of the swing tire, the tire swing, or the swimming in the pond. All those things. Our sense of smell is our one emotion that is most closely related to our memories, because the olfactory bulb and the olfactory tract bring that sense of smell information straight to the limbic system. So it's the one that's most closely associated with our emotions and with memory. Notice that's important from a biological standpoint. If you look carefully, and I will use the highlighter again to emphasize this, there is a little red dot located at the bottom of the limbic system here, known as the mammillary body. That's weird that I'm talking about that. I don't see that on the list of telencephalic features. If it's not on the list of telencephalic features, why am I talking about it? Give you a hint. Look a little further on your list? Maybe under the diencephalic features. What do you notice under the diencephalic features? There you have the mammillary body. The mammillary body is a structure in the diencephalic features. And since we're talking about it, we'll say what it does right now. I see there's a question, but let me finish this and I'll answer it. Mammillary body helps to play a role in taste and smell aversion. Right? My wife is my favorite example of this. She went to school in Southern California, and there was a restaurant down there that had this peppercorn ranch dressing that she absolutely loved. And when she was pregnant, for the first couple, for the month and a half of being pregnant, we probably ate at that place 4 days a week, lunch or dinner or something, because she loved that peppercorn ranch dressing. Then one night we had it, and the next morning she woke up with really bad morning sickness and threw up all over the place and threw up a fair amount of that ranch peppercorn. Was it the ranch peppercorn dressing that made her sick? No. But she associated throwing up with the taste and the smell of that peppercorn ranch, and guess what happened? We never ate at that restaurant ever again. All right? Or maybe you had that bottle of tequila that you ate. Well, it sometimes can go away and it sometimes can't, right? Maybe, there you were, right? Middle school as an 8th grader, and you got a hold of that gallon-sized jug of Jose Gold tequila, right? And halfway through the night, you ran out of lemons trying to use those as chasers, so you switched to oranges. And of course, after finishing off that bottle, and you were horribly sick the next day, and now the smell of an orange or even the feel of the pulp of an orange makes you nauseous. Can that eventually go away with time? Yeah, but sometimes it takes years and years, and sometimes it never goes away, right? Again, this is a life instinct. If you're a sheep rummaging through a field and you eat some red berries and you get really sick from eating those red berries, do you ever want to eat those red berries again? No. And so we have this taste and smell aversion, and that's what the mammillary body does to us. And if you look closely, what you will notice is out here is the hippocampus. Remember, where we convert short-term memory into long-term memory. Out here is the mammillary body. And if you notice, there is this yellow structure that connects the two together. This yellow structure is a bundle of axons that connects the mammillary body to the hippocampus. And that structure is called the fornix. Here in the three-dimensional view, we see it, but another place where we see it really, really nicely is in a sagittal section. Here in the sagittal section, this is a structure we'll talk about in just a minute called the corpus callosum. But this right here, this structure right here, and let's again highlight that. This structure right here is the fornix. So this structure right here is the fornix. Notice we see it on this image of a sagittal section. But let's go back here. Not to this, but to that. Notice here as well, this structure right here, that is the fornix. So on our half-head chart, on our models, on our illustrations, this structure is called the fornix, and it's easy to find because the fornix connects to this big comma-shaped structure, and this big comma-shaped structure is called the corpus callosum. Here's the corpus callosum. Notice if we go back to that other image, we see that same orientation. Here is the corpus callosum, and here is the fornix. And notice both are white because they're axons. They're bundles of axons. The fornix carries information from our mammillary gland, which is down here, to our hippocampus, which is lateral, which is why we don't see it. The corpus callosum we see in one other picture. The other picture that shows it nicely is this one. The corpus callosum is the largest white matter structure that connects the two hemispheres. Remember, we called that a commissural tract. Again, a fancy way of just saying axons that go from one hemisphere to another. All right, and that's that corpus callosum. This corpus callosum is important because it helps to coordinate the activity between the two hemispheres. So like I said, if you're going to clap, your left hand needs to know what your right hand is doing, so we need to communicate to each other. But again, this is one of those instances where understanding, knowing, let's undo that, hold on, I want to just erase this. Understanding how the body works helps us to manipulate it to our advantage. How many people here have heard of a condition called epilepsy?
Yes.
Yeah, a fair number of you have, absolutely. Typically, what happens with epilepsy is there is a region of the brain that typically part of the cortex that produces a big, huge, massive, what they call neural storm, where what happens is we get a massive production of action potentials. And that massive storm of action potentials spreads. It spreads across the cerebrum, across to the other side through the corpus callosum, and can affect the entire other side of the brain as well. And sometimes when these neural storms are big and bad enough, these people can have massive grand mal seizures where they drop to the ground and they're shaking violently, or if you don't get that wooden spoon in their mouth, they could bite their tongue off. It is an incredibly debilitating physiological process to have take place. And clearly, if you could have one of these grand mal seizures, you can't drive a car, you often can't sit in a classroom, you're very limited by what you're being able to do. The good news is that often these neural storms are produced from just one location of the cerebral cortex. Now, they can't go in and destroy that cerebral cortex because there's too many complications that could come from that. But what they can try to do is try to limit the spread. And the way they do that is by splitting the brain. What they will do is they will actually make an incision down the longitudinal fissure where they cut the corpus callosum in half. They call these people literally split brain people. Right? It's split brain is this condition where they do that. Then what happens when this neural storm starts, it's still going to spread over one hemisphere, but it can't spread to the other. And it dramatically decreases the strength of those seizures, and often they can live a normal life. Great question. Sometimes fast flashing lights can cause those type of epileptic seizures. Those types are brought on by a different type of condition than epilepsy, typically, those types of seizures. But it is a similar type of seizure, yes. Absolutely. Of course, I say these people can lead normal lives. If you've got your corpus callosum cut, can you really lead a normal life? If the left side of your brain can't talk to the right side of your brain? You would think no, but the answer turns out to be yes. And the reason for that is object permanency. Where something is in the real world is where that thing is in the real world. And when you're trying to grab your phone with your right hand and trying to grab your hand from the left hand, it's in the same place. And so even without being able to communicate, excuse me, they're actually pretty good at being able to function. In fact, really the best way to tease this out is incredibly artificial conditions. Some of them can actually drive. Yes, some of these people are actually able to drive after this happens as a result of it. Where you can see the deficiency sometimes is where COVID-19, But one of the places where you sometimes see the difference is similar to that. So exactly, great question. So someone said, what about that left side deficiencies thing? So here's what happens. Here's how you can tease this out. What they will do is they will have a person look through a viewer. And when you look through a viewer, they will look at an image. And the image that's on the viewer, on the left side will be male, and on the right side will be female. And so what they'll do is they'll ask them to write down what they see and to say out loud what they see. And what'll happen is when they write down with the one hand, they'll write down male and they'll say female, and they won't realize that there's a difference. So it's literally one of those cases where the right hand and the left hand don't know what they're doing. But surprisingly, these people are able to actually get along pretty well. No, it doesn't affect vision. And remember, I said the corpus callosum is the largest commissural structure, but it's not the only. There are a couple of other structures that cross, and so it doesn't cut all of the information. There is still some communication from the left and right, but it is the biggest structure that connects the left and the right. And like I said, shockingly, these people can live fairly normal lives, even with that deficiency. All right, we've done fornix, we've done corpus callosum. While we're here, we can see one more. Notice below the corpus callosum is a thin membranous structure. I'll highlight it here. Notice there are some spaces. One of the things we talked about is our brain has fluid on the outside, but it also has fluid on the inside as well. And these fluid-filled spaces are called the ventricles. As you can see here, there is a left and a right ventricle, and they're separated by a thin piece of membrane that separates the two. And this thin piece of membrane that separates the two together is the septum pellucidum. Here in a coronal section, we see this really nicely. The other place we see this, and we have to use our imagination a teeny bit with this one, is to come back to this picture here. Notice again, this here is our corpus callosum. Spell that right? I always get that one wrong. Callosum. Oh, that's what I did. Oh, well, corpus I just butchered. Corpus callosum, which is that structure up here. And then remember, I'm just going to put an F here because I'm running out of space. The fornix is this little structure right here. Notice in between the two of them, this right here, and let's highlight it. This right here is that membrane that is the septum pellucidum. If you think about it, if we were to poke through this, then what would happen is we would be in the lateral ventricle. If you think instead back to this view, basically we've cut, that picture is a picture where we've cut the brain in half. So all of this over here is gone, right? We have the corpus callosum up here, the fornix is underneath it here coming up. And so again, if we were to have taken our arrow and poked it through the septum pellucidum, then we would have been in the lateral ventricle. So again, it doesn't have a function. It's just a barrier between the lateral ventricles. So we see it nicely on this image here, and we see it nicely on this image here. So this is that dividing structure that is the septum pellucidum. All righty. Excellent. One last structure, and we are done with our limbic system. And for that, we need to come back to this picture. Notice here, there are three structures: the caudate nucleus, the putamen, and the globus pallidus. And the good news is you don't need to know any of those names, right? Those names are not on your list. Oops, I went too far. You don't need to know any of those individual names, but these three structures together form a special group, and these three structures together form a special group called the basal ganglia. Now, if you remember, a ganglion is a collection of cell bodies outside the central nervous system. This is inside the central nervous system. So, not surprisingly, because anatomists hate us, when they named it wrong. The good news is it is getting better. More and more in the literature, you see it called what it should be called, which is the basal nuclei. But you will still see basal ganglia in the research. So what in a lot of the literature. So I will accept both names. You may call these three things together the basal ganglia, or you may call them the basal nuclei. The basal nuclei technically is the right one, but I will accept both. And what we need to also know is what they do. In quotes, this is your autopilot. More specifically, what our basal nuclei does is it maintains a learned mechanical behavior. Oops, if I spell behavior right. There we go. Lentiform plus the caudate together. No, so lentiform nucleus, notice the, where's my pointer? Here it is. The lentiform nucleus is the putamen and the globus pallidus together. But when you take the putamen, the globus pallidus, and the caudate nucleus together, those three things together, they form our basal nuclei. Biting the nails when nervous would not necessarily, and not noticing it, oh, that's a good question. No, that would not be an example. What this is doesn't initiate an activity or maintain an activity. Or terminate an activity, it maintains it. Think of it this way. When that 1-year-old is first learning how to walk, it requires every ounce of his concentration. You can almost see the smoke coming off of his head as he's thinking about where he has to balance and where he has to put his arms and where he has to put his legs as he's putting into it. Now, you can walk and talk and eat a sandwich and tweet and text and do all sorts of other things at the same time and not think about the activity. You don't start spontaneously walking for no reason at all, but once you start walking, you don't have to maintain that activity. Or one of my favorite examples, sort of like that, we'll get to that one in a second, knitting, right? Now that, yes, the lentiform nucleus and the caudate nucleus make the basal nuclei. However, as I said, you don't need to know the terms lentiform, you don't need to know the terms putamen, you don't need to know the terms globus pallidus, you don't need the terms caudate. On the exam, what I will do is I will draw a circle around these three structures. And I will ask you, what are these three structures collectively known as? And you will say basal nuclei. And I will ask you, what is the function of these three structures collectively? And you will say that it maintains a learned mechanical behavior. Like knitting. Now that we're all stuck at home, right? Grandma on Zoom is teaching you how to knit. All right, and while you're sitting there, every knit, every purl requires all of your focus and concentration. Whereas grandma can watch TV, she can be, baking a pie, she can be having a conversation, and her hands are whirling away knitting that sweater. Again, grandma doesn't just spontaneously start knitting for no reason. But when she starts that behavior, that learned behavior is maintained. We take advantage of this all the time, and sometimes it catches us. Driving is another example of one of those things that required all of your attention when you first learned to do it. Now we're doing our makeup and eating a sandwich and tweeting and doing all sorts of other things in the car. And that's the problem, because my autopilot kicks in and I'm halfway to work and I was supposed to be going to the store to get milk. Right? My autopilot kicked in and I was halfway to work before I realized that I had taken a wrong turn somewhere. Or you're in the shower, right? And as you're in the shower bathing, right, you suddenly realize, did I wash my hair? because your autopilot had kicked in and you were going through the motions, thinking about whatever you were thinking about. If you're like me, you think about one direction while you're in the shower, and then suddenly you don't know if you washed your hair or not, right? Because that autopilot kicks in. And that's what we're talking about. Playing a piano is another great example. Playing the piano is a learned behavior that is maintained by the basal nuclei. Again, they don't start spontaneously playing the piano, but once they start playing, they can finish a song, and often they finish that song without even thinking about it, because it's a learned behavior. When you're learning it, it doesn't work. If you're just learning to walk, if you're just learning to play the piano, if you're just learning to drive, your autopilot can't kick in. All right? But once your autopilot kicks in, then it can maintain that learned behavior. All right? Questions on that? All right. With that, we are done with all of our telencephalic features. We do have to talk a little bit about our protections of the brain, and those include the fluid and the fluid-filled spaces, but we need to finish the rest of the anatomy before we get to that. And I think I've been talking for close to 4 hours now, so I think this is a good stopping point. We've gotten through the telencephalon. As you can see from the list, we've done most of the heavy lifting. There's just a few things left. A lot of that is the spinal cord, which we already talked about in the histology. So I will briefly be going over the anatomy of it again. So make sure you look at that on there, but we'll also do that in lecture as well. It's a great question. So someone asked if stuff like taking a drive or playing a piano are often done to relieve stress. I don't know if that's necessarily related to the function of the basal nuclei. It may be one of the advantages of it, but again, it's one of those things where it kicks in so you don't have to give your cognitive thought. So like I said, when you're driving, you can be thinking about other things. If you're just focusing on the driving and trying to drown out all the other noise, then that's certainly not the basal ganglia. If your autopilot kicks in while you're driving and now you're able to think of all the stress that is going on in the world right now, then I'm not sure that that's the escapism you were looking for either. So it may actually fight against the stress relief of those activities if your autopilot kicks in and then you're able to think about the stressful stuff as opposed to focusing on it. So in some ways, maybe doing something you don't know as well, like trying to learn to knit, right? Although the frustration with your lack of success could be bothersome as well, but it definitely gets your mind off of whatever it is that may be stressing you at the time. So I'm not sure. If it, on the other hand, lets your mind wander to think of other things, like one direction, then yeah, that definitely could relieve stress. All righty. I'm pooped. I don't know about the rest of you. Hopefully that's good. I think we've made some good headway on our anatomy here. And like I said, most of the heavy lifting is done. The rest of the cephalons don't have nearly as much information. And then the spinal cord we'll be talking about also when we talk about the spinal cord stuff, which is what we'll be talking about in the next class. All right. And then once we get done with all of that, I'm hoping that next class we'll also be able to do our cranial nerves as well. We'll definitely start them. I don't know if we'll get a chance to finish them. We'll see how the time goes. All right. Any questions on any of that? All righty then. I think that what I will do is I'm going to go ahead and stop the recording now. Anybody that wants to stick around and ask any kind of questions or anything like that, I will happily answer those questions there. But I'm going to go ahead and stop the recording. And so again, as always, you guys be safe, take care of yourselves, be healthy, and have fun, study hard, and I will see you Thursday. And I will stop the recording now.